Well, good day, everybody, and welcome. Thank you so much for joining us for our DOPPS Autumn Update meeting. This is timed around the time of the ASN annual meeting, and looking forward to that day, perhaps as early as next year when we can do this again in person. But until then, we really have a packed agenda today, and so really looking forward to the 3 hours that we have together. Next slide, please. All right, this is such an important time for us, really a time of celebration as 2021 is our 25th year of the DOPPS, and we're just so thrilled. It's just been a tremendous honor, privilege to work with everybody over the course of this time frame to bring the DOPPS, to bring forward the work that we've done and all the really good work that so many folks have contributed to over these years. Next slide, please. Yeah, and thanks again to our investigators, our consortium sponsors, especially to our site investigators, coordinators, and study patients who really worked tirelessly and given tirelessly to help make this study a success that it is. Next slide. All right, and many thanks as well to our consortium sponsors. It really is through partnerships with folks such as you that really allows us to do the good work that we do. We're so appreciative for your support and partnership. Thank you. Next slide. All right, well, it really does take a village. We are now in 20 countries, 775 active clinical sites, and 34,000 patients across our 3 studies within the program, our DOPPS, Pdops, and CKDOPS, and each of those 3 studies have time set aside today to review their research and exciting new developments. Next slide. And just to remind folks here, from the very start, when Fritz, Ron, and others launched this study, these goals really stand very much unaltered. That is to capture and describe real-world practice variation, and we do so with a random, stratified random national sample of participating sites, either dialysis units or now advanced CKD clinics. And importantly, not only do we identify variation in practice, we spend a lot of time focused on identifying optimal practices, and in turn, providing lessons to the community about ways that we can improve care. And actually, I have the fortune of giving a talk next week at ASN, where I will review the actually really pretty remarkable progress in hemodialysis mortality over the last 25 years. And actually, I'll be leveraging work that our biostatistician Keith McCullough has done, but identifying some key practices that have improved over time and that really have impacted survival and helped improve survival. So yeah, please take a listen to that talk at ASN. And then this is a slide showing our publication record. We don't show this at every event like this anymore, but we put this together just this week, and we're pleased to see that we have as many publications as we do this year already. So that's a way of saying that folks are just so active, and all the work that people are putting into the study really does lead to important research output. We see we have well over 20 publications so far this year. All right, next slide. Right, and by the way, the third goal of the DOPPS program is to serve as a resource for the community. And this is incredibly important. And I still feel that there's much more, or is more that we can do in this regard. We just have so much data and so much to work with, and we really benefit from input from others. Well, I was going to note that one really important collaboration with the community is our partnership with the International Society of Nephrology, which really ramped up this year. And many thanks to Roberta Paquafijo, who's a DOPPS investigator and a leader of the ISN educational initiatives. And thanks as well to the ISN leadership for helping support this collaboration. Here's a slide showing a number of webinars and video abstracts that we put on through ISN Academy and that are available on the web. And next slide. Right, and just actually, just very recently, I, along with Fritz Port oh, sorry, before getting there, this is a survey that we put together with the ISN looking at COVID and COVID care. We have an excellent presentation in this regard from Elliot Tanner later in this presentation, which actually will focus on lower middle income countries. And here's what I was just getting to. Actually, I, as well as Ron Pizzoni and Fritz Port, we're part of a podcast that Roberto led. We're actually celebrating the achievements of DOPPS over 25 years. And please take a listen. This is available through the ISN website and podcast platforms. Which actually should be a really good listen, and you hear some nice, great stories from Fritz and Ron about DOPPS in the early days. So that was actually really fun to put together. Next slide. All right, we're busy at ASN. I think this is a total of 14 abstracts. Next slide. Also, we have Ron is speaking at a symposium on Paritis, and I, as well as Ludden Golesta, will speak at a DOPPS/USRDS symposium. I would note actually that Fritz Port is the moderator of that session, and he's the perfect person to moderate this session, talking about the OGs of big data, which my daughter's reminding me means the originals of big data. But of course, Fritz launched both the USRDS and the DOPPS as well. So kudos to Fritz. And please take a listen to the symposium in 1 week at ASN. Next slide. And as noted today, we've just got a chock full agenda. We are going to have sessions on DOPPS and then Pdops, or ISN DOPPS COVID survey, and a CKDOPS session as well. Now, for each of these, we have one advantage to doing this remotely by webinars is that we do have pre-recorded talks, and we do have time in most of those sessions for interface afterwards, so chat and discussion. Please do use the chat function with any questions that you may have, or ideas that you may have as well, even beyond the talks that you see, ideas that you might have for new questions, for new research directions. And here's instructions on using the chat feature, which you all are probably familiar with after a year and a half of COVID at this point. And next slide. All right, well, actually the first session that we're going to have today, we're actually really very excited about. Before getting into the DOPPS study related sessions, we wanted to take a step back and see where we stand with the HIF-PH inhibitors. It's actually been 2 years since the pH inhibitors, roxadustat specifically, were introduced in China. They were approved in China and then in Japan as well. So we have 2 years now of being on the market there. Next slide, please. Right, and these are data from our DOPPS study. This is in-center hemodialysis, and we're seeing roughly 4 to 5% HIF-PH inhibitor use in these 2 DOPPS countries. Now, as folks may know, I mean, these are prevalent hemodialysis patients, so not necessarily the most likely group to first be using HIF-PH inhibitors, as our panelists will speak to. So what we did with this, we actually have about a half hour for this segment. What we're gonna do is have short videos from 2 of our panelists, followed by a more open panel discussion. And once again, for folks on the line, we do encourage you to submit questions, comments through the interactive medium. And with that said, let me introduce my first speaker. And this is Professor Masayami Nangaku. Professor Nangaku is at the University of Tokyo and also DOPPS, JDOPPS, Japan DOPPS country investigator, and really well known in the HIF-PH inhibitor space. So we'll start with this video from Dr. Nangaku.
Hello, this is Masayami Nangaku from the University of Tokyo. I am going to talk about current status of HIF-PH inhibitors in Japan today. This is disclosure of my COI. HIF-PH inhibitors were developed on the discovery of the Nobel Prize. Roxadustat was the first drug approved in Japan on September 2019. Roxadustat came into the market on November 2019, followed by daprodustat and vadadustat, which were approved on June 2020. Enardustat was approved on September 2020, and molidustat was approved at the beginning of this year. We performed clinical trials of HIF-PH inhibitors in Japan. Daprodustat was effective in non-dialysis dependent CKD patients, HD patients, and PD patients in Japan. Vadadustat was also effective both in non-dialysis dependent CKD patients and dialysis dependent CKD patients. Enardustat was also effective, and we published the long-term observation of the phase 3 clinical trial. Of note, we did not observe serious adverse events in these clinical trials. This is the post-marketing survey of roxadustat, performed for half a year from November 2019. The most often observed adverse event was gastrointestinal disorder. This is a table of adverse events of specific interest. As you can see, only one case of hyperkalemia, 2 cases of cancer, and 1 case of retinal hemorrhage were reported. The cases of thrombosis embolism-related events were 84. PMDA, our regulatory agency, required a description of warning about thrombosis embolism in the package insert of all HIF-PH inhibitors in Japan. This is the post-marketing survey of daprodustat, 2 cases of retina-related adverse events, 4 cases of cancer-related events, and 6 cases of thrombosis embolism-related events were reported here. This is the post-marketing survey of vadadustat. There was no report about retina-related adverse events, 1 case of neoplasm, and 3 cases of thrombosis embolism-related events were reported. Because HIF-PH inhibitors were first approved in China and Japan, it is the important mission of the APSN to deliver the information accurately to the world. So we published recommendation of proper use of HIF-PH inhibitors. Let's talk about the cost. The Japanese government decided that the cost should not be a factor that decides the choice between ESA and HIF-PH inhibitors. So they made the cost of ESA and HIF-PH inhibitors almost equal. In Japan, the cost of drugs for HD patients are bundled. However, oral drugs are out of the bundle. Only injection drugs are bundled. Theoretically, as HIF-PH inhibitors are per oral drug, that is out of the bundle. In that case, doctors may prescribe HIF-PH inhibitors based on the economic reason. So the Japanese government decided to change the rule. If one wants to include the HIF-PH inhibitors in the bundle as ESA, they reimburse more. If one wants to prescribe HIF-PH inhibitors out of the bundle, they decrease the reimbursement. HIF-PH inhibitors are slowly replacing ESAs in Japan. The speed is not fast because Japanese doctors are replacing ESA with HIF-PH inhibitors deliberately. Thank you for your attention.
Okay, yeah, Professor Nangaku, thank you so much for that that introduction. So interesting to note, first of all, that there were 5 HIF-PH inhibitors on the market and available in Japan. Number 2, to see the safety data that you reviewed briefly, and also to hear the movements with respect to cost and sort of neutralizing it, as it were, any cost incentive for or against prescription. Anyway, much more to talk about. Our next speaker is Professor Li Zhuo, and Li is a good friend of ours and adopts China country investigators from Beijing at Peking University People's Hospital. Thank you, Professor Zhuo.
Dear colleagues, I'm happy to have the opportunity to share real-world experience in HIF-PHI use in China mainland. As you know, because HIF-PHI have multi ways in promote red blood cell production, it is hoped that it's better than ESA in renal anemia management. So you know the drug was validated in non-dialytic patients and dialytic dependent CKD patients and is marketed in the year 2018 as class 1.1 new drug. This drug was covered by national medical insurance in the last year in 2019. And some patients with renal anemia changed gradually from ESA to roxadustat. And roxadustat was more favorable in non-dialytic patients and peritoneal dialysis patients. It is estimated that around 60 to 70% newly diagnosed patients started roxadustat after initial assessment. But most hemo patients was reluctant to change from ESA to roxadustat because it is easy to use ESA intravenously or subcutaneously by our nurses during dialysis sessions. But most ESA non-responders, non-dialytic or dialytic dependent, they have to change to roxadustat. And when they change to roxadustat, most of them respond well to roxadustat and reach their hemoglobin target. As you know, DOPPS were performed in China major cities, including Beijing, Shanghai, and Guangzhou. So after national medical insurance covered in the 2019 November, we see that the percent of hemo patients who are using roxadustat increased gradually and reached 5% in May this year. And in my center, the percent of patients who are using roxadustat is about 50% in hemo patients and 70% in peritoneal dialysis patients. We know that I also want to share that some new HIF-PHI is under registration trial in China. We see here some are in phase 3, some in phase 2, and most of them in phase 1 clinical trials. I hope this new HIF-PHI drug, when they market it, can add new drugs for our doctors to use to treat anemia. And during the usage of roxadustat, doctors have some feelings when using this drug, and some of the drugs was under clinical trials, and some need to be validated to design new trials. The first is that dose of roxadustat is closely related to CKD stage in non-dialytic and dialytic patients with residual renal function. And the initial dose of roxadustat is lower than the recommended doses. ESA non-responders may respond well to roxadustat. Altitude may influence the efficacy of roxadustat. The patients who live in a high altitude may need less dose. Oral iron supplement may be efficient after changing from ESA to roxadustat in some hemo patients with stable hemoglobin. And after patient change from ESA to roxadustat, we see good blood pressure response. And roxadustat may increase the sensitivity of ESA. All these are doctors' feelings when using this drug, And some of these findings are under clinical trial to be validated. Let's see the first. What is the proper dose of roxadustat when changing from ESA to roxadustat, or when roxadustat is the first renal anemia treatment patient drugs? And the second is, can roxadustat be used to increase ESA sensitivity? And this need to be a new trial to validate. Now, what is the influence of the degree of inflammation on roxadustat efficacy? So this ongoing trial. Does roxadustat have less impact on blood pressure compared with ESA? The observational studies say yes, and further controlled trial need to be conducted to validate. What is the target of iron stereo in roxadustat-treated patients? This study need to be designed and conducted. And does roxadustat increase duodenal iron reabsorption? So this study is completed and data is under analysis. And does roxadustat has impact on red blood cell lifespan? Though this study is published recently, maybe last month. And does roxadustat slow the progression of CKD? Does this study need to be designed and conducted? Does roxadustat decrease cardiovascular mortality? Because the previous study, the pooled analysis of published data gave confusing result. I think that a randomized trial need to be conducted to validate, to further investigate this question. And does long-term use of roxadustat do good or do harm to our patients? I mean the mortality and quality of life, because you know that roxadustat can promote multi-gene, can activate multi-genes. So this question need to be further addressed. So thank you for your attention.
Professor Jones, thank you so much. All right, lots of interesting material there. First of all, the number of questions you identified that we still need to answer, and also the sort of clinical experience today, some really fascinating items that you brought up. What we'll do right now is move over to our panel, and if folks could put on their cameras on the panel, that would be wonderful. So in addition to Professor Zhou and Nangaku, we have Roberto Pequafio, and also Alex Cases and Professor Cho as well. And the moment, there we go, good. Anyway, so Professor Cases is from University of Barcelona and Hospital Clinic at Barcelona in Spain, DOPPS country investigator as well. Zhangyi Cho is at Kyungpook National University Hospital in Daegu, South Korea. And Roberto Pequafio is a DOPPS investigator on our team and has been very closely involved with the HIP-H inhibitors during development. This is important, we brought these folks together because actually all of these countries, with the exception of the US, now have approved HIP-H inhibitors, with recent approvals in Korea and of roxadustat in the European Union as well. A quite different story with the FDA and the US to date. What I'll ask is for just a couple minutes first of all, from Alex and then from Professor Cho with respect to sort of the expectation for utilization of HIP-H inhibitors as they come on market, and any other sort of high-level comments you might have. Alex.
Hello, can you hear me?
Yes.
Oh, perfect. Well, first of all, I would like to thank Bruce for the invitation. And first, I would like to express my gratitude to the colleagues from countries in the European Union and UK who gave me the feedback of the situation of roxadustat in their countries. Although roxadustat was approved by the EMA in August 2021, the speed of expected uptake is expected not to be uniform. As far as I know, few countries, like Germany and the Netherlands, have roxadustat on the market, while most of the countries are negotiating prices and whether it will be covered by the public healthcare system. In most countries, the availability of roxadustat is expected during the first half of 2022, while in some countries, like, for example, Spain, we expect to have roxa in the market later in 2022. In the UK, roxadustat is licensed, but requires the approval by the NICE before being on the market.
Thanks. Good. Thank you very much. Yeah, wonderful. Yeah, Professor Cho, any thoughts on the Korean side with respect to roxadustat?
Okay, I am Chang Cho from Daewoo National University, Korea. In Korea, roxadustat passed the Korean FDA last July, but it cannot be used in clinic, still not used because the price is not decided. We expect the price will be set early next year, and it will be available at the time. In my opinion, it's more necessary for dialysis patients because we are encountering ESA resistance in these patients, but it will probably be used first for non-dialysis patients in Korea. It's my thought. Because in non-dialysis patients, the dose is probably smaller than dialysis patients, so the price can be set with relatively small burden of reimburse. So we can use that patient first. If non-dialysis patient can be administered orally without injection, the significant demand exists in patients with especially long-term follow-up period. And I do not have any information about the price of roxadustat, but regarding the prices, as Nangaku showed in their slide, it seems that it can be used. It can be used enough if a similar prices to epoetin or NASP. Although there is a problem of pill burden, it is possible to overcome ESA resistance by taking it only on the day of dialysis, and it can be prescribed to dialysis patients. Korea also applies bundle pay for dialysis costs, but oral drugs are not included in the pay, like Japan. So it is necessary that the cost of roxadustat should be included in the bundle pay, like ESA. Okay.
Pardon me. Yeah. Thank you very much. So not yet available, of course, working on pricing and reimbursement issues, but should be by sometime next year. And it sounds as if you anticipate moderate demand in a fair bit of interest in utilization of those the drugs as they become available. Good, thank you. I would want to turn, and you noted as well an issue on dosing as well, which I think is interesting. And this came up actually in Professor Zhou's talk as well. But as we think about dosing, I actually wanted to pivot first to Roberto Pequafijo for some perspective on where we stand in the U.S. and in particular what the FDA might have thought about, had feedback and guidance from FDA and what they might be expecting in terms of additional data with roxadustat and other, and then I'm thinking of other HIFs as they may come up to for FDA review.
Right.
Thanks.
Yeah, thanks, Bruce. Yeah, so by mid-July, there was a presentation from FibroGen and AstraZeneca of their full trial program, to an external independent panel of advisor, and a very interesting discussion about the data. There was identification of some potential relationship between side effects and target hemoglobin, and also the dosing of roxadustat used in the trials, and rate of rise of hemoglobin, which was raised by the panel of advisors. But really, the final response was actually received by the companies from the FDA just a few weeks ago, and that request additional clinical trial data and not exploration of the current data available. And at the moment, both FibroGen and AstraZeneca are evaluating the next steps. I think we haven't really heard what the final decision would be, but definitely new clinical trial data will be needed for the resubmission of the process. We also heard that there will be a an advisory committee meeting later in the year for vadadustat, which already presented their phase 3 trials in a couple of New England Journal of Medicine papers a few weeks ago. And finally, for daprodustat, there will be the presentation of the main results of the phase 3 trials will be presented next week at the ASN in a late-breaking clinical trials session. And we already heard the top-line results for daprodustat 2. But in terms of regulatory process for daprodustat, I'm not aware of any movement yet.
Roberto, yeah, thank you. Yeah, it's so interesting, isn't it? Really very different stance to date on the regulatory, with respect to the regulatory decisions in the US versus many other locations in the world. So we're likely to see a situation where drug will have been on market in some cases for a couple of years before coming here to the US. So anyway, with much to learn from data outside the US moving forward. For Alex, did you have some additional context regarding what you might think, what do you think with respect to roxadustat uptake once available?
Well, I think that according to the EMA assessment report and the results of the phase 3 trials, first eligible patients will be those with chronic kidney disease not on dialysis, as well as PD patients or incident patients on dialysis. However, according to the EMA, switching a dialysis patient stable with a current ISA to roxadustat should be evaluated carefully, considering the risks and benefits because of a tendency to an increased risk of cardiovascular events and mortality in this setting, especially among those that require higher ISA doses. Because of the lack of evidence, also patients with cancer or adult polycystic kidney disease cannot be considered as good candidates. With respect to reimbursement challenges, I know that in the Netherlands and Germany, roxa is available, as previously mentioned, and fully reimbursed in general. The expectations in Europe will be that they will be fully covered by the public healthcare systems, perhaps with limited indications in some countries. And for sure, price will be an issue, especially when cheaper biosimilars of ISA are available in most countries. We have to wait, I think, for the pharmacoeconomic studies that are on board, including not only direct cost of the drug, but also, for example, the sparing effect on IV iron.
Good, right. Yeah, thank you, Alex. Thank you, Alex. And with this in mind, and any questions remaining with respect to safety, of course, there's a lot that's still unknown with drug with this last recently coming on market. For Masayomi, Professor Nangaku, could you just give a comment with respect to the thromboembolic events that you reported in the post-marketing safety to date? Are there any surprises that you see here?
Yes, thank you, Bruce, for the question. I don't think the number is great, considering that many patients were taking roxadustat during the post-marketing survey. The issue of the post-marketing survey is that we do not exactly know what kind of numbers of the patients were taking the drug, and there's no control. But actually, the estimation is that probably about 20,000 patients were taking roxadustat during the post-marketing survey. So 84 thromboembolic events does not seem too many to me. Thank you.
Good. Okay. Right. Exactly. Actually, we're running fairly short on time, and Masayomi as well. Any concern or with respect to the question been raised about rapid rise in hemoglobin? Are you seeing any concerns about this in relation to safety events?
Well, thank you, Bruce. The speed of the increase of hemoglobin is completely different among 5 different types of drugs, and roxadustat increases hemoglobin very rapidly sometimes. So we are paying special attention to the too rapid increase of hemoglobin when we use roxadustat.
Yeah, indeed, right. And this, I think, that's so interesting. But speaks specifically to comments from Professor Zhou as well, who noted that they're often using lower dose than labeled. And even the FDA, I think, brought this up as an option as well. So it may be a dosing issue on the Roxa side. And any other, for Professor Zhou, any comments in this regard with respect to dosing of roxadustat? Oh, sorry, Lee, right now you're muted. Okay, sorry, Lee, we're not able to hear you, unfortunately.
Bruce, while we're waiting for Lee on this, may I just add, because this was a discussion that came up in the FDA AdCom. There was a presentation from the companies at the AdCom showing sub-analysis of data or data stratified by dosing and also by target hemoglobin, and there was an idea to expand that analysis using the existing data as a potential way of mitigating the risk observed in trials, and which was not accepted ultimately by the FDA, which requested new data. But there was the idea of really making an exercise in terms of the reanalysis and also observational data that could provide information that would answer that question.
Good. Right, Roberto, thank you very much. We're actually just past time, and let's see, Professor Zhou, it sounds as if your audio is not on right now, and that's totally fine. What we can do is take advantage of our chat function. So we actually have a number of questions that came to us that we've just not had a chance to get to. We knew as we put this session together that we would be short on time, and actually may think about ways to come up with a new session in the coming months where we bring you all together again and discuss this in more detail, because I think we really have an important opportunity here with the DOPPS data and involvement in multiple regions around the world as the HIF-PH inhibitors are coming on board. Really an important opportunity along with expertise that you all bring. So again, we'll continue the discussion through chat function and welcome comments and questions from audience as well. With this, let's move now to our next segment. All right, this is now pivoting to our section on the DOPPS, our in-center hemodialysis study. And I'll just quickly note that Ron Pizzoni, DOPPS distinguished research scientist with Arbor Research, will moderate this particular session. So Ron, please.
Thank you very much, Bruce. And that was a really great session. I enjoyed it so much. Yeah, so here's an update regarding countries in DOPPS 7. We have 17 countries, 423 sites, and nearly 12,000 patients in the US, Europe, GCC, and in Japan and China. See here. Oh, next slide, please. Yeah, thanks. Our study in DOPPS really could not be possible without the great leadership from our country investigators. Each of the countries of DOPPS right from the very beginning, and so we just wanted to again to call attention and thank our country investigators for all of your contributions to DOPPS. You play such an important role in leading the study within each country and providing such important and key feedback regarding dialysis care in your country. And this last session, I think, is just a really good example of that. All of the new information that's coming out and the different perspectives that you provide. So just want to really highlight the important role and our great thanks to all of our country investigators who play such an important role. Also, in the last couple of years or so on, we've we've increased our efforts to have DOPPS data analyzed by data hubs where there is expertise in carrying out epidemiologic kinds of analyses and the statistical kinds of expertise that one needs. And right now, with regards to DOPPS data, we have 3 such hubs. One is in Canada, based at the Ottawa Hospital Research Institute, led by Dr. Manish Sood, who's a country investigator from Canada, and in China at Dr. Li Zhuo's place at Peking University People's Hospital, and in Japan at iHope International, which is based in Kyoto in close collaboration with the JDOPPS Steering Committee. We also have a couple of analytic projects going on where we've provided data for those, and one of those is on community racial composition and outcomes, led by Dr. Laden Golestana from Montefiore Hospital in New York, and the other is a study of comparative effectiveness of beta blockers being led by Dr. Manish Sood in Ottawa. Next slide, please. So we have 3 really great, very interesting talks for this session. And the first one will be given by Dr. Angelo Caraboyas, who's a great colleague at Arbor Research. And Angelo is one of our research scientists and does such an excellent job in leading so many projects. And so Angelo, Angelo is going to speak today on comparing MBD management using etalcalcetide compared to cinacalcet, particularly PTH control. Thank you.
Hi, I'm Angelo Caraboyas. I'm a research scientist with the DOPPS team, and I'll be speaking today on real-world effectiveness of etalcalcetide versus cinacalcet in U.S. dialysis patients using a facility calcimimetic preference approach. So first, some background. Secondary hyperparathyroidism is common in U.S. dialysis patients. PTH levels greater than 600 have been associated with mortality in prior observational studies. Calcimimetics are often used to control high PTH levels. There's essentially 2 options. Oral cinacalcet is susceptible to challenges of self-management and adherence, whereas IV etalcalcetide has been shown to be superior to cinacalcet in a randomized trial setting, but real-world comparative effectiveness is yet unknown. So some U.S. dialysis facilities switched from cinacalcet to etalcalcetide as the primary calcimimetic therapy after etalcalcetide was introduced in 2017. So what we did was leverage this variation in facility calcimimetic preference to compare PTH control in those facilities that transition patients to etalcalcetide, which we call etal-first facilities, versus those that continue to use cinacalcet as the primary calcimimetic therapy, which we considered to be Cinefirst facilities. We used US DOPPS data, and for the exposure, we considered facilities to be etal-first if they treated over 75% of their calcimimetic users with etalcalcetide, Cinefirst if they treated over 75% of their calcimimetic users with cinacalcet. Those in the middle, in that gray area, were considered to be no clear preference. And then we also excluded patients with very low calcimimetic use overall. Our primary outcome was PTH, but we also looked at albumin-corrected calcium and serum phosphorus levels. We took 2 different approaches here. One was a between facility, a cross-sectional comparison, where we looked at facility calcimimetic preference, which we defined in 2019, and then looked at PTH levels in the subsequent 6 months. We compared those PTH levels in the etal-first versus Cinefirst facilities. But we also took a more longitudinal approach, a within facility pre-post comparison. And we looked at HD facilities during 2 periods. The first during 2016, when all facilities were Cinefirst before ethyl calcitide had been introduced, and then also in 2019 after the introduction of ethyl calcitide. And we looked at PTH levels assessed after the exposure period in 2016, and then after the exposure period in 2019. And we looked at the pre-post differences in PTH for switch to ethyl facilities and remain Cinefirst facilities. Essentially, did PTH levels change in those facilities that switched over to ethyl calcitide? And then we'd use the facilities that did not switch over as a comparison group to perform our difference in differences approach. This figure shows the temporal trends in facility calcimimetic type from 2017 through February of 2020. What you see at the beginning here is that all of these facilities are orange. This represents 100% cinecalcet, with the denominator being all patients prescribed a calcimimetic. You can see the uptake of ethyl calcitide in 2017 and starting to become more common in 2018 into 2019. You see these blue HD facilities. These are the centers who now prescribe ethyl calcitide to all or the great majority of their calcimimetic users. And then the orange, dark orange being still 100% cinecalcet, light orange being mostly cinecalcet. So you can see our comparison of interest here between essentially the blue facilities versus the orange facilities. The gray facilities in the middle represent those who had between 25 and 75% mix of ethyl calcitide and cinecalcet. They had no clear facility preference. And so inclusion of these centers would likely bias our results by indication if the decision to prescribe patients ethyl calcitide versus cinecalcet was mostly based on patient indication and not on facility preference. And then the facilities on the bottom, you can see, had very low calcimimetic use overall, so they were also excluded. Table 1, we show patient and facility characteristics for Cinefirst and ethyl-first facilities. You can see that the major difference here is what jumps out is dialysis organization size. And you can see that the Cinefirst facilities are mostly the LDO, MDO, the large and medium dialysis sized organizations, whereas those that switched to ethyl-first tended to be more the small dialysis organizations and independent facilities. You can see also other differences in practices, including more calcimimetic use overall in the ethyl-first facilities, and also less active vitamin D use overall in those facilities. Patient characteristics, the ethyl-first facilities had patients that were a little older, and other small differences that largely were a product of the difference in dialysis organization size. Here's our approach 1 results. This is the cross-sectional analysis. So you can see that comparing the 45 ethyl-first versus 67 Cinefirst facilities, when we looked at the calcimimetic users in those facilities, the users in the ethyl-first facilities had much lower PTH levels. You can see 479 versus 715 in terms of the mean. There was quite a bit of differences in those patient characteristics that we observed in the prior table. Once we accounted for those, the adjusted difference between them was about 115. Similarly, when we looked at the proportion of patients with PTH greater than 600, the proportion was 44% in Cinefirst facilities versus only 21% in ethyl-first facilities. Once we accounted for differences, the difference was a little smaller, but it was still an effect size of adjusted difference of 11% between the two facility types. When we looked at calcium and phosphorus, we saw much lower effect sizes, if any. When we looked at the longitudinal approach, approach 2, what we see here, when we compared 32 facilities that switched from Cinefirst to eTailFirst, and looked at 34 facilities that remained Cinefirst from 2016-17 all the way through 2019-2020. When we looked at the calcimimetic users in the switch to eTailFirst facilities, you can see PTH levels in those facilities decline quite substantially from 671 down to 484, whereas in the facilities that did not switch over, their mean PTH levels slightly increased from 632 to 698. Once we accounted for differences in characteristics, the adjusted difference was in mean PTH was 169 lower for the switch to eTailFirst facilities. Similarly, for PTH greater than 600, the proportion decreased substantially within those centers that switched over. That dropped from almost in half, from 40% to 20%, whereas the proportion over 600 remained around 40% in the Cinefirst facilities, with an adjusted difference of about 14%. Again, you see calcium levels slightly decreased. The proportion with calcium less than 8.4 slightly increased, but certainly not to the extent that we observe for PTH. So to summarize, we leverage variation in US HD facility practice to conduct this sort of natural experiment. Following introduction of etalcalcitide in 2017, we isolated those facilities with a clear calcimimetic preference so that whichever drug each patient was getting was more likely determined by the facility preference and not by patient indication. We observed better PTH control in those facilities that switched over their calcimimetic prescriptions to etalcalcitide versus those that continued to use cinacalcet as the primary calcimimetic therapy. Calcium levels were slightly lower in the eTailFirst facilities, and phosphorus levels didn't seem to be associated with facility calcimimetic preference. So despite advantages in efficacy shown in trials, in real world effectiveness shown here, etalcalcitide use does remain limited, largely due to the cost environment in the US. Though we looked at MBD markers and levels here, but note that further research is needed to investigate the impact on clinical outcomes. Thank you.
Thank you very much, Angelo. It's really an excellent presentation. And I just want to remind folks that if you have any questions, to please submit them using the Q&A feature on your screen, and we'll try to address questions after the end of the session, after all three speakers have had a chance to present. So our next presentation is by Dr. Pablo Urena, who is one of our country investigators from France. And actually, this is part of a project that is with our DOPPS investigators. Every year we try to carry out a research project, and this is one of those projects that was selected. And so Pablo, thank you very much for giving us a presentation today, which we're really looking forward to. Thank you.
Hello, ladies and gentlemen. I'm Pablo Antonio Urena Torres. I am working as the Chief of Dialysis Service at the Aura in Saint-Ouen, Paris, France. Together with Professor Christian Combe and Fitsum Gebre, we are the principal investigator for DOPPS in France. Before starting, I would like to thank the DOPPS team, in particular Angelo Carabollas and others, for allowing us, Naïde Tabizadeh and myself, to do this study. The study title is The Diuretic Prescription and Incident Hemodialysis Patients International Practice Pattern. As you know, there is not too much data regarding the use and the dosage of diuretics in chronic hemodialysis patients. And in some of the already published studies, it has been suggested that the continuation of diuretics in this patient could reduce the risk of hospitalization and mortality. To get more information and probably to help better inform international therapeutic strategies, we'll here investigate the regional variation in the loop diuretic use and dosing strategies in association with clinical and dialysis-related outcome. We here present the very preliminary descriptive data of the DOPPS cohort analysis. For that, we examined the DOPPS cohort, which included more than 7,000 patients during the DOPPS phase 2 to 5 from 2002 to 2015. That group of patients included 27,759 patients with a dialysis vintage less than 1 year, what we call the incident dialysis patients. Then the parameters we analyzed were the following: diuretics use, yes or not, according to the dialysis vintage; diuretics, yes or not, regarding the type of diuretics among the incident dialysis patients; the percentage of diuretic use by facility in the same patients. That was restricted to the facility with more than 5 eligible patients. And then, because there were several types of loop of diuretics, we converted the 20 milligram of torsemide equal to 80 milligram of furosemide, and 1 milligram of butamide equal to 20 torsemide or 20 milligram of furosemide. You can see here that the most frequent type of diuretic used among the country was the loop diuretics, 95 and 96%. Only 3 to 4% of these patients were using other type of diuretics. Now, this slide shows you the evolution of the patient or the percentage of patients taking diuretics during the first 5 years of dialysis. And you can see that the percentage of patients decreased constantly and progressively from the first year to the 5 years of dialysis. For example, here in Germany, in the black line, you see that it goes from 85% at the beginning to less than 40% after 5 years of dialysis. It was the same for the other countries, like here in France too, also in Italy from 60% to 20%, and even in countries using less than 30% of dialysis were taking diuretic at the beginning, and only 5% were taking after 5 years of dialysis. Next, this slide illustrates the large variation in the use of diuretics within country. 70 to 80% of patients in Germany and Sweden were taking diuretics during the first year of dialysis. 60 to 30% in Belgium, Canada, France, Japan, and only or less than 20% in countries like the country of the Gulf, Spain, UK, USA. There was also a large variation in the doses of diuretics within country. You see here that the mean doses of diuretics in Germany and Sweden was around 500 milligrams a day. That in United States, UK, Canada, and other countries, the dose was lower than 200 milligrams a day. So we can say that there is a substantial international variation in the diuretic prescription practices among incident dialysis patients. The next steps we are planning to do is to explore association between the diuretic use, doses, and one, the volume parameters like blood pressure medications, the weight gain, the residual urine volume, and also the clinical outcomes like hospitalization, mortality, MACE, heart failure, the incidence of fractures. We are also planning to collect the Pdops, the physical and mental scores, the vitality energy, the recovery time from each hemodialysis session. We are also planning to look at the bone and mineral disorder markers like calcemia, phosphatemia, PTH, total alkaline phosphatase, and also changes in the electrolyte disorders like bicarbonate, sodium, and potassium. We are expecting to get the preliminary results in the first Q1 2022. Thank you very much for your attention, and I will be delighted taking questions.
Thank you very much, Pablo. This is a really interesting presentation, and boy, the variation across countries and facilities is really amazing. So now we'll progress to our third presentation in this session. And this one is being given by Dr. Nidhi Sukul of the University of Michigan, who is a great colleague who we've worked with on many projects in DOPPS, and particularly with regards to Paritis, which is such a difficult symptom experienced by many hemodialysis patients. And so here's this presentation on some recent work on Paritis by Dr. Sukul. Thank you very much, Nidhi.
Hello, I'm Nidhi Sukul, a clinical nephrologist at the University of Michigan, and welcome to an update on self-reported Paritis in hemodialysis patients, associations with outcomes and course of symptoms over 12 months. To begin, I'd like to take you through DOPPS's recent publication titled Self-Reported Paritis and Clinical Dialysis-Related and Patient-Reported Outcomes in Hemodialysis Patients, which was published in Kidney Medicine in 2020. It has previously been shown that chronic kidney disease-associated Paritis has far-reaching effects on the lives of patients on hemodialysis. But information regarding the relationship between Paritis and several key outcomes in HD patients remains limited. So we sought out to investigate associations of Paritis severity with mortality and hospitalization, withdrawal from dialysis, missed dialysis treatments, recovery time from a dialysis session, health-related quality of life measures, and employment status. In this study, over 23,000 patients on hemodialysis and DOPPS phases 4 through 6 were included and answered a single itch-related question from the KDQOL 36, which asked, during the past 4 weeks, to what extent were you bothered by itchy skin? With response options including not at all, somewhat, moderately, very much, or extremely. As stated in the previous slide, we investigated the association of Paritis severity with clinical outcomes, dialysis-related outcomes, and patient-reported outcomes. This figure demonstrates the prevalence of CKD-AP internationally, displaying the distribution of Paritis prevalence and severity here among many countries. Looking at moderate to extreme Paritis, which is the green section down, we see that the prevalence was notably highest in the United Kingdom at 47% and lowest in Germany at 27%. And when we looked at the outcome of mortality, we saw that extreme Paritis was associated with higher hazard of all-cause and cause-specific mortality. Compared to patients who reported being not at all bothered by itchy skin, patients who were extremely bothered had a nearly 25% higher rate of all-cause mortality. Patients extremely bothered by itching also had nearly a 30% higher rate of cardiovascular-related mortality and nearly a 45% higher infection-related mortality. The association with cardiovascular mortality may relate to higher levels of inflammation or possibly the higher prevalence of heart failure seen in these study patients. And the infection-related mortality may relate to derangements in the immune system or the presence of a central vein catheter. Indeed, those with extreme Paritis had the highest prevalence of catheter use in this study, which may serve to partly explain the higher infection-related mortality seen among those extremely bothered, given the high risk of fatal infections seen in patients with dialysis catheters. With regard to rate of hospitalization, the adjusted rates of all-cause, cardiovascular-related, and infection-related hospitalizations were all about 20% greater for patients extremely bothered versus not at all bothered by itchy skin, while the rate for skin-related infections was about 40% greater, and the rate for mental status change or confusion-related hospitalization was nearly 85% greater, though the precision of these estimates was limited by the low number of events, and so should be interpreted with caution in the setting of this small event rate. It should also be noted that this study adjusted for 15 comorbidities, so these associations are not likely to be explained by the overall illness of the patient, but potentially linked to central nervous system disturbances associated with pruritus or its treatment. Patients with extreme kidney itch were 1.5 times more likely to withdraw from dialysis, which may be a manifestation of the contribution of pruritus to already significant symptom burden in patients on dialysis. Patients with extreme kidney itch were more likely to skip dialysis sessions, which also may be a reflection of the high symptom burden among these patients. Patients with extreme kidney itch were less likely to be employed, and the cumulative symptom burden may also play a role in the difficulty sustaining or seeking employment. Patients were also more likely to experience a prolonged recovery time after a dialysis session, with increasing severity of kidney itch, potentially adding to the psychological burden of kidney itch, and this may contribute to missed treatments or eventual withdrawal from dialysis. It has been shown in previous work, this study showed that progressively more severe pruritus was strongly and monotonically associated with progressively poorer self-reported mental and physical health-related quality of life, as measured by the physical and mental component summary scores, where lower scores indicate poor quality of life. Progressively more severe pruritus was also associated with self-reported depression, as measured by the CESD-10, self-reported restless sleep, being bothered by faintness or dizziness, and being bothered by feeling washed out or drained. Our findings demonstrate how diverse and far-reaching poor outcomes are for patients who experience CKD-AP, specifically those with more severe pruritus, and underscores the need for change in practice patterns to effectively identify and treat patients with pruritus, to reduce symptom burden and improve quality of life, and possibly even survival. This next abstract to be presented at this year's ASN conference is titled Pruritus in Hemodialysis Patients, Course of Symptoms After 12 Months. We have now seen that the burden of CKD-AP in HD patients has been well established, including its association with adverse clinical events and patient-reported outcomes. And while prior studies have focused on a single baseline CKD assessment, longer-term studies of CKD-AP severity over time are limited. So in this study, the aim was to quantify the extent to which pruritus symptoms vary over 1 year for individual patients. In this study, over 8,000 HD patients across 21 countries and DOPPS who had 2 CKD-AP assessments, 12 months apart, were included. The variable of interest was CKD-AP severity, as assessed by the single question asking about the extent patients were bothered by pruritus in the preceding 4 weeks, with the same 5 response options. Responses were stratified at month 12 by baseline responses, and then we estimated the proportion of patients whose pruritus symptoms worsened, improved, or stayed the same. The proportion of patients at least moderately bothered by pruritus was 37% at baseline, 37% after 1 year, and 51% at either assessment. 28% were at least very much bothered by itchy skin at either assessment, stressing the chronic nature of this symptom. Overall, 43% of patients provided the same response 1 year later, compared to 28% whose pruritus improved and 28% whose pruritus worsened. After 12 months, the majority of patients exhibited relatively stable disease. Among patients not at all or somewhat bothered by pruritus at baseline, 22% became at least moderately bothered 1 year later. Among patients at least moderately bothered by pruritus at baseline, 60% remained at least moderately bothered 1 year later. Our findings suggest that at least half of chronic HD patients were affected by CKD-AP over the course of 1 year, reflecting an unmet medical need. CKD-AP symptoms remain unresolved 12 months later for the majority of HD patients bothered by itchy skin at baseline, demonstrating the chronic nature of the condition. Future research will examine the association between within patient changes in pruritus symptoms and changes in other PROs. Lastly, we would like to understand the severity and temporal changes in CKD-AP in HD patients via frequent EPRO collection in DOPPS, and here the data collection is ongoing. Currently, we are carrying out prospective EPRO data collection at DOPPS Europe sites at quarterly intervals during an 18-month data collection period, yielding up to 7 serial measurements of pruritus and other PROs. Planned analyses include CKD-AP and PROs, evaluating approaches to measure CKD-AP burden by comparing various CKD-AP instruments and their associations with PROs, longitudinal changes in CKD-AP symptoms, describing within patient patterns and their association with PROs, and CKD-AP and short-term outcomes, including healthcare resource utilization, hospitalization, and mortality in the 3 months following CKD-AP assessment. Lastly, I wanted to put in a quick plug for a book that is to be published in February 2022, authored by Dr. Hugh Rayner and myself, titled Fast Facts, Kidney Itch, in which we address the fact that kidney itch is common, but often undiagnosed, and even when recognized, it is typically inadequately treated or not treated at all. In it, we describe the problem of kidney itch in detail, document the impact it has on people's lives, review the current understanding of its pathophysiology, and set out an evidence-based approach to its assessment and management. Currently, it is freely available as a course through Karger, for which CME credits can be obtained. This 3-part interactive course highlights the impact of chronic kidney disease-associated pruritus on patients' daily lives with advice on evaluation and holistic management. I have put the link to access this course at the bottom of this slide here. Thank you very much.
Thank you very much, Dr. Suko. And I must say, I've had a chance to read the Fast Facts Kidney course that Dr. Hugh Rayner, who is one of our emeritus CI country investigators from UK and NITI have created, and it's absolutely excellent. Such a great resource, and I think many of you and your colleagues would find it very useful if you have a chance to look at it. So there's a few questions here, and thanks to all 3 of you for your absolutely excellent presentations. And one of them is for Pablo, which is, and I see that I think many audience members that would be interested in this, and that in the US, a lot of nephrologists are not so comfortable in giving such high doses of loop diuretics to their patients. And do you see that urine volume increases much with using such high doses? And do you have any safety concerns in that regard?
This is a very good question, Ron. And you know, there is a lack of data. And what I can say is very subjective, because we haven't measured the hearing capacity of patients receiving these high doses here in Europe, because we are giving up to 1,000 milligrams a day. And it appears that this patient keeps a greater urinary volume for a long time. But I cannot say that there is no probably safety issue, but we don't see an alert or big side effects. But everything is speculative. I don't have strong and solid data to confirm that.
Yeah. Oh, thank you. Yeah, it was amazing to see the differences across countries in use of the loop diuretics. I think it ranged from 30% to about 85% among patients on dialysis less than a year, and across facilities within countries. It's just huge. It's just a really
huge. And I hope this data will change the practice and the recommendation from KDIGO, because there is nothing in this field.
I was curious if you've had a chance to, and those are based off of DOPPS 2 to DOPPS 5 data, basically 2002 to 2015. Have you seen much change over time, over that time period, in use of loop diuretics? Have you guys had a chance to look at that yet?
No, Angelo, probably you can tell us, but I don't think there is big change between the DOPPS phases regarding the percentage of, patients using diuretics.
Yeah, we have looked into that, and we have not found a big a big difference over time in the loop diuretic prescription.
Okay, well, great. Thank you, Angelo. Nidhi, I was really taken by the findings on changes in over 1 year on how patients, to what extent they're bothered by uremic pruritus. And I was wondering if you've, and this is the first time we've ever looked at this over such a long period of time, and if as a clinician and your experience in this field, have you found those results to be surprising, or that fits pretty much with what your expectation is? And even patients who didn't at the beginning weren't really bothered, and then about 22% were moderately to extremely bothered a year later, and so on. I was just wondering what your, what your impression has been of these first results of, over the longitudinal changes in pruritus.
Yeah, absolutely. Thank you so much, Ron. I think they're not too surprising. Oftentimes when we have patients with pruritus, as they get treated, I see that they sometimes will have an initial improvement, and then as, I don't know if their body just gets used to the therapies that we're trying, the pruritus tends to kind of creep back. And so the fact that many patients are still suffering from it 1 year later, I don't find too surprising. And then what I have seen mostly in my clinical practice is that patients who have been stable, seemingly without pruritus initially, eventually start to develop some pruritus. And this doesn't happen with every patient, but seeing as what I'm seeing in our dialysis patients, it does not surprise me that we do see some patients over time develop the symptom.
Oh, great. Yeah. Well, thank you so much. That was such an informative presentation. And Angelo, just one question for you, because I know we have to get moving on to the other sessions, is that in the work that you did, in some facilities, cinacalcet is given within the dialysis unit 3 times a week instead of daily, taken at home daily by the patient as is recommended by the label. And I was just wondering to what extent, if you had a sense of what percentage of patients in your analysis were taking cinacalcet thrice weekly as opposed to daily.
Yeah, it's around half, maybe a little under half of the cinacalcet users in the U.S. who are getting it 3 times a week at the dialysis session.
Okay, and that really hasn't been examined, has it? The efficacy of thrice weekly versus daily, and so I guess that will be, but it is certainly important. Looks like it's a major part of practice here in the U.S.
Yeah, there's been some small single center studies that have compared the 2 practices, but certainly nothing on the scale of DOPPS that I'm aware of.
Okay, well, thank you. I just want to thank all 3 of you for such excellent informative presentations and lots of exciting work going on. So yeah, thanks so very, very much. Okay.
Thank you.
Thank you, everyone.
Everybody. Bye-bye.
Bye-bye.
Yeah, thank you all, and thanks, Ron. That was a great segment and really nice questions and answers at the end. We are so interested in particularly what's issues important to patients, and this does include, as Pablo's highlighted, residual urine volume, critically important to quality of life, but also pruritus, as Dr. Supel spoke to, and other topics. By the way, the residual urine volume diuretic project was result of a competitive submission from our DOPPS Europe investigators for analytic projects of high importance. And we do have another call for such proposals coming up at this before the end of this year for next year. So we look forward to this. Well, we'll move on now to our peritoneal dialysis segment, Pdops, and the great fortune of working so closely with Jeff Pearl, who will be the moderator for this segment. Jeff is the principal investigator for Pdops and works at the University of Toronto and St. Michael's Hospital in Toronto. And Jeff is just an incredible leader in the clinical science of peritoneal dialysis. And perhaps even more important, a tremendous advocate for peritoneal dialysis and doing it right and doing in the right sorts of patients. So we're so fortunate to have Jeff working very, very closely with us. Jeff, take it away.
Thanks, Bruce, and hello, everybody. It's a pleasure to be speaking here today. And, you know, the irony here is we're all sort of doing this from home because of COVID-19. So there certainly have been benefits to receiving dialysis at home. And I'm so delighted to see peritoneal dialysis featured today as part of the DOPPS program of research. So this is a Pdops phase 2. These are our country participation, and we've had 236 sites with over 11,000 PD patients. And again, this is a real testament to all of our colleagues and friends around the world who have been able to collaborate and really do amazing work to get us to where we are. And in terms of the country investigators, this is our list of country investigators. And again, I just want to take this opportunity to thank everybody for all of their input and all of their leadership in the Pdops study. In terms of partners in research, similar to the other aspects of the DOPPS program, we've been developing data hubs, and there's 3 such data hubs in Toronto, in Australia, and in the United Kingdom, all doing analyses with the Pdops data, in addition to the wonderful analyses that are being done at Arbor Research. And there are also local study headquarters, including Chulalongkorn University in Thailand, led by our Thai investigators, and in South Korea. And we expect, you know, Thailand has been exceptionally productive in making the study relevant, and we look forward to continued output from South Korea. In terms of some ancillary studies, there's been several ancillary studies funded. You'll hear about our infection work in terms of the OPUS study coming up, and also studies looking at PD catheter function. So with that in mind, the Pdops session will first start off with insights into PD-related infections from the US and beyond. And it's a pleasure to introduce Eric Young, who's been working very closely with the US claims data to try and look at how to ascertain infection rates in the United States. And I'll be presenting some data from the Pdops study. Hello, everybody. It's a pleasure to present to you insights into peritoneal dialysis-related infection from the US and beyond. And I'm joined by Eric Young, who's a senior scientist at Arbor Research, and we'll be presenting you some of the latest information from our sub-study, Optimizing the Prevention of PD-Associated Peritonitis in the US. Next slide. So just want to remind everybody, our investigators in the OPUS study, and want to thank them for all of their participation. And it's been really fantastic to work with such amazing individuals. And support for this pilot study is provided by a grant from the AHRQ. Next slide. And so I just want to remind everybody what our study aims are for OPUS. One of our study aims is to develop a standardized definition for peritonitis and a protocol for peritonitis reporting. To use this definition to understand the variability in peritonitis rates and outcomes, to identify patient and facility characteristics that impact the risk of peritonitis, and clinical practices that impact the risk of peritonitis, and as well to work with knowledge users, including patients and providers, to understand how we carry out these aims and how do we knowledge translate these practices. Next slide. So in terms of one of our aims of standardized peritonitis reporting, next slide, we've had applied the ISPD definition, and here's the ISPD definition for peritonitis: 2 of the following: abdominal pain or cloudy effluent, a cell count of greater than 100 with 50% neutrophils with a dwell time of at least 2 hours, and a positive culture. And taking that definition, next slide, and collected information, next slide, in a prospective way across sites in the US. And this was in response to a review that we did of the literature, suggesting that when we analyzed randomized controlled trials and other studies that had published information on peritonitis, different definitions of peritonitis were employed. Next slide. So using a standardized definition developed by our investigators and in collaboration with the definition from the ISPD, we are prospectively We are prospectively collecting information on at 62 study sites on 190 peritonitis episodes reported to date. And this is a one-year pilot to say, can we as a PD community and clinics come together to standardize and collect peritonitis in a standardized fashion? Next slide. This is the sites that are participating, and I want to thank all of the sites, including some of our dialysis organization partners, for participating in the pilot study. Next slide. And these are our key findings. You can see here, at least when we submitted this data to the ASN, we've had 86 peritonitis episodes. And one thing, just to provide some insights in terms of tracking peritonitis in a standardized way, is a very high rate of culture negative. And you can see here, 21 cases of the 86 were culture negative peritonitis, suggesting that this is a larger challenge than we previously thought. And as the study comes to the end, we'll be sharing more insights about what we found in the tracker pilot study. Next slide. Looking at the Pdops data and what we've learned about peritonitis rates, practices, and outcomes. Next slide. We've learned that there's huge variability in peritonitis rates across the world, and even within clinics, there's significant variability in peritonitis rates. And in the Pdops data, there was an overall peritonitis rate of around 0.28 episodes per patient year. Next slide. One of the things that we've also learned is that uptake of evidence-based practices is quite poor in some countries. In the US, for example, only 63% of medical directors endorsed using antibiotics at the time of PD catheter insertion, which is a grade 1A ISPD recommendation. Next slide. Next slide. And so when we looked at the risk factors and factors that were associated with a lower risk of peritonitis, you can see here using antibiotics at catheter insertion was associated with a 20% reduction in the risk of peritonitis. And that might be the practice itself, but it also might mean that clinics that adhere to evidence-based practices may do so in other realms of peritonitis prevention. Interestingly, we also found that greater use of APD was associated with a lower risk of peritonitis. Next slide. One other important point that has come through is that culture negative peritonitis was a major challenge, both in our tracker study, as we showed you, and also in Pdops. Next slide. And we understand that according to ISPD guidelines, less than 15% of episodes should be culture negative. And there are ways to reduce that in terms of how the effluent is handled. Next slide. When we look at our culture negative peritonitis rate, you can see that it was well above the ISPD recommendations in many countries and ranged between 14 to 28%. It was particularly high in Thailand. Next slide. And if we look at the impact of different organisms on outcomes of peritonitis, and this is a composite of catheter removal, death, transfer to hemodialysis, you can see here that culture negative peritonitis tends to be associated with a lower cure rate compared to gram-positive peritonitis as the reference group. Next slide. Next slide. Oh, and so in this quality improvement project within Thailand, they noticed this as well. And they sent samples not only to a local lab for analysis, but also sent samples to a central lab as a quality improvement project to reduce culture negative peritonitis. Next slide. And what you can see here is that what the central lab, if we go back, grew many more organisms than were grown locally, suggesting that locally, microbiology labs in Thailand may not be adhering to best practices when it comes to optimal culturing of the effluent, because the central lab using ISPD endorsed practices did yield more organisms than was at the local lab, which reduced the risk of culture negative peritonitis. So a really important quality improvement project. Next slide. If we look at hypokalemia, one of the papers that was published in our experience was the relationship between low potassium and peritonitis. And there is a relationship, but once we adjust for patient characteristics, including nutritional factors, that attenuates the relationship. So in part, the relationship between low potassium and peritonitis may be mediated by markers of nutrition that may track with a heightened risk of peritonitis. Next slide. If we look at peritonitis and infection, it's the leading cause of transfer to hemodialysis across all countries in the Pdops, suggesting that we need to develop strategies to prevent it And once peritonitis has developed, we need to develop strategies to reduce the risk that it leads to an adverse event. Next slide. And so when we look at those adverse outcomes following a peritonitis, you can see here, this is the distribution of deaths, hemodialysis transfer, catheter removal, and relapse and recurrent peritonitis, which are all adverse events following a peritonitis. And you can see the distribution of these across countries with some subtle differences between the countries. Next slide. When we looked at the cure rate by country, which was a composite of not having a relapse or recurrence, not having the catheter removed, not transferring to hemodialysis, and not having a mortality event following peritonitis, we can see that there is variability within countries, and again, some subtle differences with cure rates across countries. Next slide. Pointing back at this graph, you can see that not only was culture negative peritonitis associated with a lower rate of cure, but gram negative, typically bowel bug peritonitis, was associated with a lower risk of cure compared to gram positive peritonitis, suggesting that these might be very difficult infections that we need to improve cure rates from to reduce adverse events following a peritonitis event. Next slide. One of the interesting findings here was that when centers were using an aminoglycoside or ciprofloxacin-based empiric gram-negative antimicrobial as their empiric therapy for peritonitis treatment, there was a much higher rate of cure. And perhaps it relates to emerging resistance with cephalosporins or higher antibiotic levels with the uses of aminoglycoside and ciprofloxacin that made these potentially superior in terms of resolution of gram-negative peritonitis. An important finding in Pdops that needs to be confirmed in other studies. Next slide.
I'm going to talk about using Medicare claims to understand PD infection rates, outcomes, risk factors, and modifiable practices in the US, or at least some of those topics. Next slide, please. By way of background, peritonitis is obviously an important problem, as Jeff has illustrated. And prior studies, many of them may have a relatively small or non-representative sample. And as we've heard, there are sometimes variable definitions applied to defining peritonitis. And so we thought that Medicare claims might provide a unique population-based perspective featuring large sample size, a more representative sample, and that it has thus far been an unexploited opportunity for studying PD-associated peritonitis. So we use the USRDS standard analysis claims files from 2013 to 2017. These are files with inpatient, outpatient, and physician supplier claims. We identified those ICD-9 and 10 codes that were used to identify peritonitis. We reasoned that closely spaced claims were considered part of the same peritonitis episode, and we defined a new episode to occur when there was a new claim that was greater than 30 days from any prior peritonitis claim, or more than 50 days from the initial peritonitis claim, in the absence of any other claims in the 10 days before that time point. Next slide, please. So this just shows the variability in diagnosis codes. You can see that we had in the range of 70 to 80,000 claims per year. So overall, we analyzed over 375,000 claims. We identified 28 ICD-9 and 10 codes. We switched from ICD-9 to ICD-10 in this time period. We identified 28 codes that seem to indicate peritonitis. Rather than showing all 28 of them, I'm just showing the categories that we identified. The first one is our codes that we call acute peritonitis codes that simply indicate peritonitis in some sense. There were 9 such codes, and they account for the majority of claims, 71% in 2000 13 down to 61% in 2017. Over all years, they accounted for 65% of claims. So this is the biggest category, although as you can see, it is declining over time. Then we identified 2 codes that indicated spontaneous bacterial peritonitis. These codes are usually used for patients with liver disease and ascites and are probably not necessarily appropriate for patients with PD-associated peritonitis. Nonetheless, they are used about 3 to 6% of the time, 5% overall, and we think they actually indicate PD-associated peritonitis. We identified 13 codes that we called complication codes, usually due to a perforated viscus, appendicitis, intestinal perforation, something of that sort. Those accounted for about 2% of claims. And then finally, there were 4 codes that we've called catheter codes. These signify inflammation or infection of the catheter and or peritoneum. And it's a little ambiguous as to whether these codes are for exit site infections or for peritonitis. We actually think they are used to indicate both diagnoses. These codes are used 28% of the time, and their use is actually increasing over time. So overall, these claim codes we applied our rules, our timing rules, and they yielded over 70,000 episodes. There was a mean of 5.6 claims and a median of 2 claims per episode. 40% were exclusively outpatient, 9% were exclusively inpatient, and 16% were exclusively based on catheter code claims. Next slide, please. And this shows the rates that we found. So we had a reference set of rules that I've partly described. And using these rules, we found an overall rate of 0.54 episodes per patient year, with actually a decline from 0.61 in 2013 and a steady decline to 0.49 in 2017. So it was falling by about 5% per year. Now, our sensitivity analyses looked at other ways of defining peritonitis. We've done several such analyses. In one, we excluded the use of catheter codes altogether. Remember, this is that ambiguous category where we're not sure if it's indicating an exit site infection or peritonitis. When we exclude these codes, we find an overall rate of 0.45 episodes per patient year. And then when we restrict episodes to those where there's at least one claim that was submitted by a dialysis facility or a nephrologist, we find a rate of 0.35 episodes per patient year. And this gets us more in the realm of what is reported in dialysis facilities and what Jeff reported for the Pdops study. And it suggests that clinic reported rates may have an issue, and that the clinic may not be aware of all the episodes of peritonitis that may happen in hospital settings or outpatient settings where the dialysis facility may not be involved. That's a hypothesis that needs to be explored further. Next slide, please. And we found that peritonitis rates varied by year, as I've said, and by patient characteristics. So here's a couple of graphics that show peritonitis rates on the y-axis and year on the x-axis. The panels on the left show the impact of various demographics. First, you can see age, that younger patients tend to have a higher peritonitis rate in any given year. You can see there's a significant race effect such that Black patients have a higher peritonitis rate than white patients, and Asian patients have a lower peritonitis rate. You can see there's a vintage effect. In these crude unadjusted analyses, patients with longer vintage have a higher risk of peritonitis. And then finally, there's an impact of ESKD cause, such that diabetic patients tend to have higher peritonitis rates. Now on the right, we show the impact of prior peritonitis episodes. And when there's no prior peritonitis episodes, the peritonitis rate is in the range that we've talked about. But with each increment increase in the number of prior peritonitis episodes, there's a very large, substantial increase in peritonitis risk. You can see the impact of 1, 2, 3, and greater than 4 prior episodes of peritonitis. So that's an important risk factor. The next slide shows a multivariate Poisson model of peritonitis risk where we combine these various factors. And I'm just going to show selected factors that were in the model. We actually did 2 models, Model 1 and Model 2. Model 1 has patient demographics without including the number of prior peritonitis episodes. And you could see the age effect that I talked about, younger patients having a higher rate. You can see the race effect, higher rates in Black patients, lower rates in Asian patients. You can see the cause of ESKD impact, diabetes, and to some extent hypertension having a higher risk, and the vintage effect or time with ESRD, risk going up with higher vintage. But in Model 2, where we also include the number of prior peritonitis episodes, you see similar age, race, and cause effects, but the vintage effect changes. It turns out the higher vintage patients also are more likely to have prior episodes. So when you account for that, which is again a very big effect, you can see that the vintage effect goes away, and in fact, the higher risk is associated with patients with a lower vintage. So there's a really dramatic interaction between vintage and prior peritonitis episodes as illustrated in this multivariate analysis. Next slide, please. So to summarize our initial work on using claims, we found considerable coding heterogeneity that we think indicates a lack of standardization among providers. And there is a need for clearer coding guidance so that we have more consistency in use of diagnosis codes. We found that peritonitis rates varied from 0.35 to 0.54 episodes per patient year, depending on the rules used to derive episodes from claims. And these rates are a little bit higher than what is often reported by dialysis facilities and other studies. We found that peritonitis rates declined by about 5% per year between 2013 and 2017. We identified some initial risk factors. There are more to explore, but in our initial analyses, we found an impact of age, race, diabetes, and prior peritonitis. And overall, the findings raise important opportunities that we hope to explore, including reconciling these findings with clinic-based measures of peritonitis rates. We want to promote uniform coding standards. We think there should be some discussion of modernization of diagnosis coding to capture all the aspects of PD-associated infections in a better way. And we think that this approach will lead to help with efforts for targeted practice improvements. And we do think that this claims-based approach offers a robust framework for the study of PD-associated peritonitis. Thank you.
Thank you, Eric. So some insights from CMS claims data, some insights from the Pdops international study, and some insights from tracking peritonitis across facilities with a standardized approach. Thank you for your attention, and so look forward to questions. Thank you. Our next presentation will be regarding a new initiative with Pdops, the DOPPS Practice Monitor with insights into real-time PD care, presented from Jenny Shen, Assistant Professor of Medicine at UCLA Harbor.
Hello, my name is Jenny Shen, and today I'm here to talk about the DPM Goes Home, insights into real-time PD care. A little bit of background, there are 780,000 individuals in the US who receive dialysis or have a kidney transplant, with nearly 60,000 patients dialyzing at home with PD. The PD population is expected to increase, so it's important to know what clinical practice trends are looking at. In terms of the DPM-PD, it is the DOPPS Practice Monitor Peritoneal Dialysis, and it's a web-based report of up-to-date trends in clinical care of US PD patients. The reports include clinical practice and PD prescription data with more than 150 charts, figures, and data tables. This data comes from the Pdops study, which includes 150 US dialysis PD facilities that together contribute data on over 4,000 PD patients. We should note that facilities and Pdops have to have at least 20 PD patients, so it may not be reflective of trends in smaller dialysis units. The DPM PD data are updated every 6 months to reflect the most recent changes in clinical PD practices, which may be due to payment or policy updates, availability of new products, or other reasons. And to access it, you can visit this website, www.dopps.org/dpm-pd. When you click on that link, you're going to come to this webpage, and it'll show DOPPS Practice Monitor Peritoneal Dialysis. And up here, it should say PD. This is actually a drop-down menu, and you'll see that you can also pick HD because there's also a DPM HD that's been around for some time. You can play with that drop-down menu, and you can compare the data between the PD and the HD populations. If you look on this first tab here, Browse All Data, the first thing you'll have to choose is if you want to filter by clinical topic, and in future iterations, you can also filter by facility or patient characteristics. You then choose a category, and these are the categories that are available: demographics, nutrition, diabetes, cardiovascular, PD prescription, anemia, PD urea clearance, potassium, residual kidney function, and mineral and bone disorder. If you click on this tab on the far right on About PDPM, it reviews the data that we talked about before in terms of the Pdops study and where the data are coming from. Now we go back to this first tab and browse all data, and you filter by clinical topic. And let's say you decide to choose demographics. You'll see there's a number of different graphics that are going to come up under demographics. And the first one of these is age and categories, looking at the national sample. So what this means is you look at all the different time periods from April 2019 to August 2020, and it'll tell you the distribution of age of the patients who were in the Pdops study at that time. So you can see here that over time, from April 2019 to August 2020, that the average age of PD patients may have gone down somewhat, because you look up here in the dark blue, this is the percentage of patients who are at least 75 years old, and that has seemed to shrink a little bit over time. Now, in future iterations, you will be able to stratify by facility size, Black race, and time on PD as well. So for example, you could look at the distribution of age of patients by race. Now, going back to demographics, if you want to take a look at the race of PD patients in Pdops over this time, you can see that it's stayed pretty much steady here. About 25% of patients on PD have been Black. Now, if you compare that to the DPM HD or hemodialysis, you can see that the percentage of patients who are Black in hemodialysis is far higher, excuse me. So you can see here that there's fewer Black patients who are on PD. In terms of diabetes as a cause of ESRD, you can see here that maybe about 45% of PD patients report diabetes as a cause of their ESRD. Now you compare that to the hemodialysis data. In the hemodialysis population, there's a higher percentage of patients who have diabetes as a cause of ESRD. So the prevalence of diabetes as a cause of ESRD is definitely lower among patients on PD than among HD in the DOPPS studies. If we look at time with end-stage kidney disease, again, we see that over time, as PD has gotten more popular, the time on ESKD has slightly shifted down. So if you see here in the light blue, this is the percentage of patients who have been on end-stage kidney disease, have had end-stage kidney disease for only about 0 to 3 months, and has actually gone up over time. So this might represent more incident patients starting on PD. As we look at some trends now in mineral bone disorder, we'll start with PTH. You can see that the distribution of lab values hasn't really changed a whole lot over time. It's fluctuated a little bit, but it is notable that overall, about 20% of patients have a PTH that's more than 600. And similarly, if you look at serum phosphorus, again, the distribution of labs hasn't really changed a whole lot over time. But you can see that about 15% of patients have a phos above 7, which is pretty high. In terms of anemia, if we look at hemoglobin here, again, the general trends in the lab values hasn't changed a lot. You have about 20% of patients who have a hemoglobin below 10 and about 20% of patients who've got a hemoglobin above 12 here. Now, we take a deeper dive into the anemia data. You can actually take a look at trends in ESA use. It hasn't really changed very much. And the EPO use in PD patients, I would note, is lower than it is among hemodialysis patients. Now, as we look at ferritin here, the ferritins have slightly trended up. So if you look here in the green, these are the patients who have a ferritin that is at least 1,200, quite high. And you can see that it's now about 20% of patients who have that very high ferritin. On the other hand, if you look at transferrin saturation, the TSATs, if you look here, the percentage of patients who have a TSAT less than 20% may have gone up a little bit over time. And finally, if you're looking at IV iron use, it's gone up a little bit, but then has come down slightly as well. So the IV iron use has gone up and down, hasn't really mirrored the trends in ferritin or TSAT. And finally, if you look at PD modality, you can see that CAPD here in the blue has tended to go down over time here. So thanks again for letting me introduce the DPM PD. We hope you all have a chance to look at it and use it. It really gives a unique opportunity to look at some real-time data that's not available elsewhere. And this will shed insight into how clinical practice in PD is evolving. Thank you very much.
Thank you. Thank you, Dr. Shen. And I just want to remind everybody, we'll do our best to answer all the questions in the chat. And as always, feel free to reach out to us after. We were not going to have time for a live Q&A, but really want to make sure we answer your questions. And our last presentation will be by Dr. Marcelo Lopez, who will talk about association of markers of mineral and bone disease with adverse outcomes in patients on PD.
This is Marcelo Lopes. I'm an analyst at the DOPPS study, and it's my pleasure to be here at the DOPPS Autumn Update Meeting 2021. And today I'll talk about mineral and bone disease in patients on peritoneal dialysis. So there's a Pdops study, and it's titled An International Comparison of PTH and Serum Calcium Control and Associated Outcomes Among Patients on Peritoneal Dialysis in Pdops. Although we know that for patients that are on hemodialysis, that out of range levels of PTH and calcium are associated with worse outcomes, we don't know that for sure for patients on peritoneal dialysis. We have previously shown an analysis using Pdops data that phosphorus, which is another mineral and bone disease marker, is associated with adverse outcomes, especially cardiovascular outcomes, the MACE, major adverse cardiovascular events. We don't know that PTH and calcium have this association. Recently, a study showed that PTH levels, especially low PTH, is associated with peritonitis. But for other adverse events, this association has not been clear. So this analysis has the following objectives: describe baseline serum PTH and calcium levels across Pdops participating countries from the years 2014 through 2021. And we also want to test the association of baseline PTH with all-cause mortality and the major adverse cardiovascular events, MACE. So for this analysis, we included patients that were on PD from Japan, Australia, New Zealand, Canada, Thailand, the UK, and the US from phases 1 and 2 of Pdops, and South Korea from phase 2 only. The 6,473 patients, they had to have at least 6 months of treatment of peritoneal dialysis treatment, since we expected that in the beginning of as they start on that therapy, there need to be some adjustments for MBD medication, and so we thought that this could confound the results a bit. So we prefer to exclude these incidents, very early incident patients. We also excluded patients that were hybrid. They will be on PD and HD at the same time. And for the MACE analysis, we only included patients that came from facilities that had good reporting of causes of death and hospitalizations, and also collected PTH regularly. Our primary exposure was the PTH level, most recent in relationship to the baseline. Primary outcomes was all-cause mortality and also MACE, which is a combination of cardiovascular mortality and hospitalizations for stroke, unstable angina, myocardial infarction, and heart failure. We censored patients that transferred to hybrid therapy or hemodialysis, transferred to a new facility, were transplanted, or were followed up until the study end. Here we describe patient characteristics based on categories of PTH levels. There are some things to point out in this table here. First of all, most of the patients are within the range of 150 through less than 600 picograms per ml of PTH. The patients that are on the high end of PTH levels, so greater or equal to 800, they are on average 10 years younger than those with the less than 50 picograms per ml of PTH. Black patients are more likely to have high PTH, so they are 4 times more likely to be on the 800, greater or equal to 800 group than the less than 50 group. And patients also have more time since they started PD in the low PTH group. So the low PTH group have patients that have been on PD for more time than the other PTH categories. In terms of dialysis prescription, I want to point out that for the patients with the low PTH levels, so the less than 50 group, they have a much greater proportion of patients that were prescribed a high dialysate calcium. So dialysate calcium greater or equal to 3.5 milliequivalents per liter. In this group, they were 3 times as likely to have this high dialysate calcium compared to the greater than 600 PTH group. In terms of country variation of the PTH levels, a few things are worth pointing out. In Japan, patients with lower categories presented with a higher proportion there compared to all other countries. The high PTH levels, they were pretty similar in the US, Thailand, and Canada, much lower in Japan. In terms of serum calcium variation across countries, a few things are also worth pointing out. Most of the patients were in the normal range. In the Asian countries, Japan, South Korea, and Thailand, there was a greater proportion of the patients with lower the 8.5 serum calcium levels compared to all other countries. In this table, we show the association of baseline PTH with all-cause mortality by level of adjustment. So in Model 1, it's basically stratified by country and study phase. Model 2, we add demographic variables, age, race, and sex, comorbidities. In Model 3, we add to Model 2 the residual kidney function, PD mortality, and PD volume. So basically, for PTH and all-cause mortality, we don't see an association, neither in the unadjusted model all the way up to a model with all covariates. In this table, we show the association of baseline PTH with MACE, so major adverse cardiovascular events. Already in Model 1, where we just stratify by country and phase, you can see that there is an association with low PTH levels, and it gets stronger as we add covariates in the model. There are no clear associations with the high PTH categories with MACE, although also this association gets stronger as we adjust. So in conclusion, patients on PD with low PTH levels, meaning less than 50 picograms per ml, they have a higher rate of major adverse cardiovascular events. As you saw, the association gets stronger as we add more confounders in the model. For the high PTH levels, we did not see that association, although as we add more confounders to the model, the effect estimates get stronger. So there could be possible unmeasured confounders. There was no association of PTH levels with death. So for the all-cause mortality, no association was seen even with adjustments. Some PTH variation is seen across Pdops participating countries. Japan has more patients with lower PTH levels. Black race is associated with high PTH levels. So they are 4 times more likely to be on the greater or equal than 800 picograms per ml than the low, less than 50 group. Patients with lower PTH levels, they have a larger proportion of high calcium dialysis prescriptions. So even though they are on the low end of the PTH levels, so less than 50, they have a much greater proportion of patients that are prescribed high calcium dialysis compared to the other PTH groups. Also, for this group, less than 50, they have a higher PD vintage, meaning they started therapy before all other categories of PTH. Due to the study design, we cannot exclude unmeasured confounding and also other scenarios that are associated with the observational nature of the study design. So this study calls for further investigation with more appropriate design to investigate this question. Thank you for the opportunity, and I look forward to hearing your questions.
Thank you. Thank you, Marcello, for a really interesting session, and leaves us with a lot of questions. And what a great way to end the Pdops session with some really interesting findings. Turning it over to you, Bruce, and thanks for listening to the PD portion of the Autumn Update meeting.
Jeff, thanks so much. Fantastic. And I think this just illustrates the importance of bringing these data to the fore, right? Really doing very serious data collection and analysis with the large enough samples of PD patients, and we have much more to learn, of course. Jeff and speakers, thank you all very, very much, and we'll move on now to. We have 2 final segments. We're just a couple minutes behind schedule, but doing okay. Well, first of all, I'll have a presentation related to the DOPPS ISN COVID survey, and then following that, move into our CK DOPPS session. And moderator for both of those will be Roberto Paguafijo. Roberto is a senior research scientist at Arbor Research and also a professor at the Pontifical University in Curitiba, Brazil, maintaining a close relationship with folks down there. Roberto also has leadership roles with the ISN as well, and is the principal investigator for our CK DOPPS study, bringing all sorts of enthusiasm and expertise to CK DOPPS. So without further ado, I'll pass things over to Roberto.
Thanks so much, Bruce. I've been enjoying this presentation so much, but now it's time to move to our next section of the program, which will be presented by Dr. Elliot Tanner. So my friend Elliot is a nephrologist and a kidney health advocate and the CEO of an NGO called Kidney Health International. He's also a senior lecturer at the Kwame Nkrumah University in Ghana. He was the chair of the Young Nephrologist Committee of the ISN and is a very active member of the Education Working Group. Perhaps most importantly, he is the leader of the manuscript based on the results of the ISN DOPPS survey on the impact of the COVID-19 pandemic on dialysis services, with a particular focus on low and middle income countries. So now let's hear from Elliot.
Thank you very much for the invitation to have a presentation on behalf of the ISN DOPPS work that we did on COVID-19, the ISN DOPPS survey. So it's a COVID-19 pandemic identifies significant global inequities in hemodialysis care in low and low middle income countries. My name is Elliot Krantintano, and I am a nephrologist in Ghana, and I'm happy to give this presentation in the next few minutes. So, yes. All right, so I have no disclosures, and indeed, by way of introduction, we all know COVID-19 pandemic has had devastating global impact on healthcare system, more especially in low and low middle income countries. And indeed, it's all because of insufficient investment in healthcare, poor healthcare infrastructure, and then low skilled labor, to name a few. Patients on HD have particularly have challenging issues, and they had reported mortalities of about 10 to 41%, basically. And indeed, all of this as a result of various factors that have been stipulated: advanced age, diabetes, hypertension, cardiovascular diseases, all these pre-morbid or comorbid conditions, impaired immune systems. And then basically in the HD unit, we believe that the poor social distancing also be a factor. Why are there challenges? So it's actually, like I mentioned, a fragile health system in low and low middle income countries, lack of diagnostic testing. So these are particularly had to do with the COVID-19 itself, low staff numbers, which has been associated with physical and mental stress, and then the strict lockdown. Indeed, some of us realized the challenges patients and even staff had during the lockdown in terms of transport, and indeed the limited access to PPEs. And this is what we hope to survey to find out. Indeed, there has been guidelines that has been set by various organizations, the CDC, the EDTA, the United Kingdom, International Society of Nephrology, and the African Association of Nephrology also, which focus mainly on low and low middle income countries. But then the point is that for you to have a successful implementation of guidelines, you actually need some resources to be available. So that was also challenging. Why? Because resources were not available. Because for successful implementation, you need the availability of resources, infrastructure. You need access to testing, indeed, and that has also been a major challenge. You need adequate PPEs for staff and then patients alike when they especially come to the HD units. And then the isolation rooms, basically for HD. So now you suspect a patient has COVID and all. I mean, how do you isolate the patient if you don't have the infrastructure to isolate such a patient? And then again, the availability of requisite human resource for optimum management of cases. Now, our research question was, has the COVID-19 pandemic disproportionately affected care in chronic dialysis patients in low middle income country settings? And what was our method? So ISN together with DOPPS came together to have this online survey to be able to answer one objective. And the objective was to describe the global inequities in kidney failure care as they relate to income status. And we use actually the World Bank classification. So what has been the journey so far? So we all know COVID-19 started in Wuhan, basically in 2019, December. We didn't think that in 3 months down the line, it would be declared in March as a pandemic. And the ISN, the initial survey was developed. So interestingly enough, DOPPS were doing their own thing, and ISN as of the time were doing our own thing. And it so happened that since we had people of the two, we came together to be able to develop a survey, because the survey was not just now for the DOPPS countries, but it had to be extended to the non-DOPPS countries. So the first stage was actually a stratified random sampling that was used basically in terms of the DOPPS methodology, where countries with greater than 40 HD units were actually asked to stratify in terms of the public and then private, in terms of geographical location. But in most countries that had less, or in some countries, or countries that had less than 40, they were actually asked to virtually select all of these countries. So what was the result like? So we sent out to about 113 countries, and indeed we had 43 only responding. That gave us a survey number of about 240, including that of the China DOPPS. So at stage 2 actually was thrown in, where a convenient sampling method was actually used, where we just sent out emails and then used the regional boards and all in terms of the ISN groups to be able to get a bit more in terms of the response. And we were able to get about extra 162 surveys from together 78 countries. So totaling, we had 412 responses from 78 countries. The distribution was basically about 4% from low-income countries, 27% from low-middle-income countries, 34 and 35 from the high-income and the upper-middle-income countries, respectively. The survey was actually divided basically into patient-level factors and then the staff-level factors. So the patient-level factors were mainly about diagnostic testing, interruption to hemodialysis delivery, restrictions to access to hemodialysis, ICU care, and mechanical ventilation, and then reported mortalities. For the staff level care, we were interested in, again, the diagnostic testing availability and a shortage of PPEs, training and guidance that some of these staff were getting, and the psychological support. So for the patient level factors, what are some of the things we found? So we clearly realized that there were unavailability or limited availability of diagnostic testing more in low and low middle income countries as compared to the high income and then the upper middle income countries, as you can see by the figure. Then for those for diagnostic testing, we also, for asymptomatic patients, realized that routine diagnostic testing was more commonly done for people in the high end. And then you realize that those in low and low middle income countries were really shortchanged. What about the turnaround time? And indeed, I can identify with turnaround time for patients when for testing for patients, because you've done a test, you are waiting for the response, and averagely was taking over 7 days to get it. And indeed, our survey also showed similar. So you realize that if you take, let's say, diagnostic testing for low and low middle income countries, majority indeed were 3 to 7 days. When you go to the high-income countries, you realize that on the same day, they are getting response. And indeed, these were challenges that we faced during the time of the pandemic. Out-of-pocket payments, like you can see from the figure, was more in low and middle-income countries. So the patients have to pay out of pocket to be tested. So what if the patient does not have money, they don't get the testing done, they don't show up for dialysis units, and probably they might portend to worse outcomes in terms of their management. And then the challenges in terms of transport, again, more challenges in low and low middle income countries as compared to the upper middle and then the high income countries. So this is actually one of the most important slides, which we realized that indeed those who had chronic kidney failure, who were on admission, who required mechanical ventilation, who required ICU admission, they were restricted because of their condition. And you realize that in low and low middle income countries, there were more restrictions. So if you look at ICU care, for instance, in blue, there were more restrictions in low and low middle income countries as compared to the upper and then the high income countries. If you take the deeper blue, which is those who were dialyzed whilst on admission, basically, again, the restrictions were more in the low middle income countries, and so on and so forth, as you can see. Mortalities reported. So these were reported mortalities. These are not mortalities that we measured ourselves. So it's actually a rate, what do you call it, a limitation. So reported mortalities were such that those who reported the dying of about 50% of patients was actually about 30 in low and middle income countries as compared to 8, so in the high income countries. So in terms of staffing, the staff level problems were such that asymptomatic testing was basically for staff was less. And indeed, this again was a problem. If you have a patient, you are exposed to a patient with COVID-19 who you are not too sure has it or does not have it, and you need to be tested, indeed, that testing became actually a problem for you as a staff. And this was some of the things we experienced in setting where your hospital will tell you, okay, just go and stay at home for 2 weeks and you just don't know what to expect, instead of probably being just tested to know what exactly was going on. So shortages of PPE was another big challenge. And indeed, this was reflected also in our survey. Low and low-income countries reported severe shortages of PPEs at the peak of the pandemic. And the use of PPEs beyond the manufacturer's shelf life, again, was more in low and low-income countries as compared to the high income countries, as you can see. Paying out of pocket for PPEs indeed was such a big staff problem. So indeed, if you really wanted to use a PPE adequately, you needed to go and buy it because it was not available. And thus, again, we can see in low and low-income countries was such a big problem as compared to the high. This survey, part of the survey was only given to those with, those with in the low and low-income countries. And we realized that, again, the low-income countries suffered less training, less guidance, less psychological support as compared to those in the low-middle-income countries. Indeed, in conclusion, we have noted that there are striking global inequities in COVID-19 management, in the diagnostic, infection prevention, and access to routine care. Higher apparent mortalities in patients with chronic HD in low and low-income countries is likely multifactorial, like it's been said, reflecting the poor access to diagnosis and care of COVID-19, as well as greater disruption of their HD care. So then there's urgent action is required if we are able to bridge the gap in terms of these inequities, because it is very important that as we get along, we'll be able to get low and low-middle-income countries concerned. So thank you very much for the time, and I would appreciate your questions as they come through. On behalf of the ISN DOPPS survey, my name is Elliot. Thank you very much.
Well, thanks so much, Elliot, and congratulations for leading this this great work and great collaboration with the ISN. I think we'll continue answering the questions through the chat, and with that, I will just move to the next and the last part of our meeting today, which will be focused on our non-dialysis advanced CKD study, CKDOPPS. So this is an overview of the current status of the CKDOPPS. We have at the moment 5 participating countries, 154 sites, and a bit over 11,000 patients. So the participating countries are listed here: US, Germany, and France, Brazil, and Japan. There is a plan for expansion of our study ongoing for clinics in China and also in Germany, in Europe. So here's a list of our study investigators from different countries. I wanted to just highlight their names in here. So Antonio, Ricardo, and Viviane for Brazil, Benedict, Christian Jacqueline, and Ziad Massi in France in collaboration with CKD. Germany in collaboration with the DM, we have Danilo, Gerhard, Helmut, and Johannes. In collaboration with Rich J in Japan, we have Kunihiro and Takashi. And the investigators from Arbor Research dedicated to the trial are here: myself, Ron Pizzoni, and Bruce Robinson. So we have expanding a list of partners in research, both for data sharing and also for the development of ancillary studies, which are all very welcome. So if you have ideas, if you have suggestions for studies, ancillary studies, or initiation of activities to share data and analyze data of our databases, please feel free to connect with us. We're happy to discuss. At the moment, there is a strong collaboration in terms of data analysis done in France with the CKD group led by Benedict Stangel, which serves as a data hub and an analytical center for many of the ongoing projects. He also recently transferred data to the University of California in San Francisco for a project on risk-based care and equity in advanced CKD. And also, there is an ongoing study going on at the New York University, led by Dr. Scher, on the description of conservative management and practice patterns in advanced CKD, which are very happy to support and collaborate. So with that, I wanted to move to the agenda today. We have 3 presentations, and the first one will be given by Dr. Viviane Calice da Silva. Viviane is a nephrologist at the Prorim Foundation and an adjunct professor of medicine at the Univille University in Joinville in Brazil. She is a CKDOPS investigator, and she's going to be talking about iron deficiency and clinical outcomes in CKD patients not on dialysis. Viviane?
Good morning. My name is Viviane Calice Silva. I'm a nephrologist from Brazil, and it's my pleasure being here today and present to you our work entitled Associations between Serum Biomarkers of Iron Stores and the Progression of Kidney Failure in Patients with Severe Chronic Kidney Disease. This is our disclosures and affiliations. And I would like to start to show you the previously reported findings from our studies, the demonstration of association of serum biomarkers of iron stores and worse physical health-related quality of life in the CKDOPS cohort. For this study, we evaluated 2,500 patients from France, Brazil, US. The exposure variable were TSAT or ferritin. Primary outcomes evaluated were HR-QOL questionnaire, mainly physical component summary and mental component summary. And as you can see here in this graphic, the PCS and MCS evaluated adjusted and non-adjusted for hemoglobin levels. And we found that those patients who had a TSAT below 15% and also a ferritin below 50 or greater than 300 had a worse PCS scores associated with these values. These findings were not confirmed for mental component summary, and this article was published at NDT this year. Also, when evaluated the functional status of these patients, we found that those patients who had TSAT below 20% and ferritin below 50 or greater than 300 had a lower functional status. Considering physical activity, those patients who had lower TSATs were less likely to perform intense physical activity. And hemoglobin, when adjusted this analysis, only slightly attenuated these observed effects that we just mentioned. Another important finding that we already published this year in JAMES is the association between iron stores and higher risk of all-cause mortality and major cardiovascular events in our cohort CKDOPS. We evaluated in this work more than 5,000 patients from Brazil, France, US, and Germany. The exposure were TSAT and ferritin, outcomes, all-cause mortality, and major adverse cardiovascular events. As you can see here in these graphics, we can find the hazard ratio for all-cause mortality and different ranges of TSAT. All the models that were performed are shown here for different numbers, the adjustments of covariates. And what we found is for all-cause mortality, those patients who had a lower TSAT below than 15% had a higher hazard ratio for all-cause mortality even after adjustments, as well as those patients who had higher TSAT levels greater than 46%, again, have a higher hazard ratio for all-cause mortality. When this was evaluated to the major cardiovascular events, only the association with the lower levels of TSAT was confirmed after adjustments for this covariate. So as you can see here in this graphic, those patients who had a lower TSAT below 15% had a higher hazard ratio for MACE. Considering that, and also the evidences in the literature that iron supplementation in patients with heart failure is associated with an improvement in renal function, and also with the fact that the impact of these iron stores on the risk of kidney failure has not previously been explored, our hypothesis with our work is that iron deficiency might lead to an increased risk of kidney failure through two different mechanisms. One, indirectly, through the cardiorenal connection with iron deficiency leading to kidney failure induced by worsening of heart function. And also a second one, directly through the effect of kidney tissue iron content leading to worsening in kidney function. We had the aim to estimate then the association between iron deficiency and progression of kidney failure among our patients in CKDOPS. The study population was 5,400 patients enrolled in the CKDOPS between 2013 to 2017 under nephrology care from Brazil, France, Germany, and the US, with an eGFR below 60 mL per minute at the study enrollment, and also with TSAT or ferritin levels available at the baseline. The outcome variable analyzed was kidney failure composite endpoint. That is the first of the 4 events below: dialysis initiation or kidney transplantation, or 40% decline of eGFR from baseline, or sustained eGFR below 15 mL per minute. The exposure variable was iron deficiency, defined as TSAT below 20%, and sub-classified as functional or absolute according to the ferritin levels. The main analyses performed were Cox proportional hazard models stratified by countries and also adjusted for a number of covariates that I will show you later on. This is our patients' characteristics by country. Here you can see that their mean eGFR and also mean TSAT is pretty similar according to each one of the countries, and the same occurs with ferritin. The median follow-up of this cohort was two years, and during the follow-up period, there were 1,800 kidney failure events, which gives us an incident rate of 15 per 100 patients a year. And this is our model results, unadjusted and adjusted. The model 1 is the crude model adjusted, stratified by countries, and the model 2 is the model 1 adjusted for age, sex, black race, BMI, eGFR, albuminuria, 11 comorbidities, ESA use, serum albumin, white blood cells, hemoglobin, and either ferritin or TSAT. As you can see here, none of the models, for the adjusted model, none of the variables evaluated, TSAT or nor ferritin, was associated with kidney failure events. So in conclusion, our previous studies demonstrated that the lower levels of biomarkers of iron stores are associated with lower physical component of quality of life and also higher risk of major adverse cardiovascular events. Levels of biomarkers of iron stores, TSAT and ferritin, are not associated with development of kidney failure in these patients evaluated, and these results support to focus attention on cardiovascular outcomes in patients with CKD. The next steps of our project is perform some additional analysis to explore whether or not heart failure modifies the association between iron deficiency and kidney failure progression, and also to estimate if hospitalization due to decompensated heart failure would accelerate the progression of kidney failure. Thank you so much.
Thanks, Vivian. That was a great presentation, an interesting project. Both the background that you showed of the published studies looking at this, it really changes the way we think about iron deficiency and outcomes in chronic kidney disease. So let's move to the second presentation. We'll leave some questions to the end. Please feel free to use the chat to ask questions to our presenters today. So now to present the piece on patirimer use in German patients, I wanted to invite Professor Helmut Reichel, who's a nephrologist and a professor of medicine at the Wien Academic Institute for Nephrology and lead investigator for CKDOPPS in Germany. Welcome, Helmut.
Good day to everybody. The current study describes patirimer pharmacoutilization in real-world German CKD patients with moderately to severely reduced eGFR. Patirimer is a non-absorbed sodium-free potassium binder that has been shown to reduce serum potassium in patients with hyperkalemia and thereby enable RAS inhibitor therapy. We will describe predictors of patirimer initiation and time to discontinuation among CKD patients using contemporary data from German participants in the CKDOPPS study. We identified 113 patirimer users with matching potassium measurement in German CKDOPPS until October 2020. And those patients were compared to more than 13,000 patients during this period who never initiated patirimer. eGFR was lower than 60 mL per minute and serum potassium was equal or higher than 4 millimoles per liter. We used logistic regression models to test adjusted comparison, and we estimated time to discontinuation of patirimer by establishing a Kaplan-Meier curve, censoring on death, dialysis, transplantation, or loss to follow-up. Results are shown on this slide. The strongest predictor of patirimer initiation was serum potassium. With rising serum potassium, the chance of starting patirimer strongly rose, and as you see here, serum potassium higher than 6.5 millimoles per liter had a more than 140-fold higher chance of starting patirimer. Another independent predictor of patirimer initiation was CKD stage, with more advanced CKD stages having higher incidences of patirimer use. No strong association with patirimer was found for gender, age, RAS inhibitor use, diabetes, coronary artery disease, and heart failure. 33% of patirimer users were already receiving alternative hyperkalemia treatment, compared to 5% of the non-users. And the use of RAS inhibitors was similar in patirimer users versus non-users, both higher or equal to 80%. The time to patirimer discontinuation is shown on the graph on this slide. We estimate that among patients initiating patirimer, less than 50% discontinue its use during the initial year. To summarize results, main predictors of patirimer initiation were advanced CKD stage and hyperkalemia. The treatment decisions to start patirimer did not appear to be based on other patient or clinical characteristics. A third of the patirimer users were already receiving alternative hyperkalemia treatment, and most patirimer users continued use of this medication for several months at least, suggesting that there is a need for longer-term control of chronic hyperkalemia using patirimer. Despite the differences in serum potassium, use of RAS inhibitors was similar in patirimer users versus non-users. Further analysis with a larger population and measurements of serum potassium before and after patirimer initiation may improve the understanding of its pharmacoutilization in moderate to advanced CKD. acknowledgement shown on the last slide. The study was supported by VIVOR, and global support was provided by the ongoing DOPPS program. We also thank the Wissenschaftliches Institut für Nephrologie from the Verband Deutsche Nierenzentren for gathering data. Thank you very much for your interest and your attention.
Thanks so much, Helmut. Wonderful to hear your presentation. We'll collect the questions on the chat box, and we'll get back to you right after the last presentation, which will be presented by Elodie Speyer. Elodie is a great collaboration of the DOPPS program, and she's an epidemiologist and has recently been appointed as the project leader for CKD study at the Center for Research and Epidemiology of Populations at INSERM in France.
So
let's hear from Elodie now.
Hello everyone. I am pleased to present you our last findings about an ongoing project focusing on barriers to conservative care from the patients and nephrologists' perspectives that we have performed into the framework of the Cicadera studies. We know that kidney failure concerns an increasing number of older, frail and dependent patients in whom dialysis may not improve the quality and/or quantity of life. A legitimate treatment option for this patient is the conservative care. And in 2015, the KDIGO Consensus Conference has defined comprehensive conservative care as plain holistic patient-centered care, including all care except dialysis. And that includes intervention to delay CKD progression, active symptom management, detailed communication, shared decision-making, and also several support resources. Nonetheless, although this treatment option may be applicable to a large proportion of patients with kidney failure, nephrologists apparently propose it rarely at notably diverse rates across Europe. For example, a 2009 survey of nephrologists across 11 European countries found only 10% reported offering this treatment option. The reason why conservative treatment remains a marginal option are unclear. Data on nephrologists' perspectives about conservative care are scarce, and those from patients even rarer. The few relevant studies have highlighted both communication issues between patient and physician with a frequent confusion between conservative and end-of-life care. It can be also explained by a lack of resources to implement this option. By using CKDOPS methodology and instrument, we investigated facility resources, nephrology practices, and patient information and their understanding about conservative care in France. The Cicadera study has been conducted in 40 nephrology outpatient facilities, nationally representative geographically and for legal status. That means public hospitals, for-profit and non-profit hospitals, where 3,033 adult patients with CKD stage 3 to 5 were enrolled. Of these, 1,361 with an eGFR less than 30 were eligible for this analysis, since we are usually discussing renal replacement therapy at that time. Questionnaires were administered to nephrologists and patients to investigate facility resources, nephrology practices, and patient information about conservative care. So, as a result, we have 38 participating facilities which have responded to the survey questionnaire, including 16 universities and 11 non-university public hospitals, and also 11 private for-profit or not-for-profit clinics. Among the 137 responding nephrologists, those in public hospitals were younger and more often women. They reported spending more time in non-clinical activities than their counterparts in private clinics, but the median duration of a routine visit was similar. Finally, about 90% of eligible patients answered to a self-questionnaire, whom 35% were older than 75. In terms of nephrology clinic resources, all reported their ability to routinely offer conservative care for advanced CKD patients. While half of university hospitals and about 30% of other facility types reported they have a written protocol or guideline for conservative care, fewer than 10% had an individual or team primarily devoted to this activity. The median estimated percentage of patients receiving conservative care was 3 to 8% according to the facilities. In the meantime, we've asked nephrologists what words do they use when referring to the care of patients with CKD stage 5 where a decision is made not to start dialysis. As you can see in this word cloud, nephrologists use many different terms. The most common were conservative management, 65% of them, and non-dialysis care with 10% of them. In regards to clinical practices regarding conservative care which were reported by nephrologists, only 29% of nephrologists have reported discussing conservative care with all their CKD stage 5 patients other than 75 years old, and most never or seldom offer it to this group. Then, 84% of nephrologists estimated that 10% of fewer of their older patients with eGFR less than 10 and uremia symptoms never started dialysis, most because they were deemed to be poor or marginal candidates. Where this option is often entrusted to palliative care team in about half the conservative care cases. Although most nephrologists reported they are fairly or extremely comfortable discussing conservative care with their patient, that means 82% to be exact, only a third reported discussing it with all their CKD stage 5 patients other than 75 years old, and most of them never or seldom offer it to this group. And even among those claiming to be extremely comfortable with this discussion, only 40% often or always suggested it to their patient. When we asked the nephrologist about which criteria would lead them to discuss palliative or conservative care as an option for their patient with advanced CKD, the 5 main factors were primarily the patient's preferences for this option, the quality of life, the frailty, the comorbidities, but also the functional status of the patient. Following these 5 criteria, they have reported social support or caregiver or physician preferences for this option. And these factors had even more influence on the decision of nephrologists who reported they are extremely comfortable with the conservative care discussion. Now I am going to talk about the perspective from the patient. From the 1,200 patients who completed a PQ at baseline, only 5% reported their doctor had informed them about a no dialysis option, and only 2% would choose it if their kidneys failed. The informed and uninformed patient did not differ significantly in their social demographic characteristics, but informed patients were, however, seen significantly more often at private clinics and tended to report a lower disease burden and more frequent attendance at an educational session than the uninformed. These results were similar in the analysis restricted to patients aged 75 years old or older, and confirmed the trend for the informed group to include a higher proportion of patients who had attended an educational session. In conclusion, this original study with a triple perspective highlights several barriers to conservative care at all levels. First, there is for sure a lack of resources in terms of material, but also in terms of staff, since all of clinics have reported to be able to routinely offer conservative care, but less than 10% of patients use this treatment option. We also identified an heterogeneous and possible and clear terminology used by the nephrologists to talk about conservative care to their patients. And for example, we know that there is a lack of training in France about that topic, and maybe this is a reason why, although 80% of nephrologists are comfortable to discuss this treatment option, there are less than 30% reported discussing it with all their CKD stage 5 patients older than 75 years old. Finally, only 5% of patients reported to have heard about conservative care, even though they were all at stage 5, 4, or 5. And we can imagine there is maybe an unclear communication between the physician and the patient, and obviously a lack of understanding from the patient sides. Thank you very much for your attention, and I am available to answer any question that you may have.
Thanks so much, Elodie. We do have a couple minutes here. We're wanted to just wait this few seconds here to get people back to the session here. I think we have Elodie and Vivian. Maybe I'll get started with you, Elodie. Interesting results. We've seen very similar findings in the US, also in Jennifer's project, which probably calls the attention to the need for like some kind of educational initiatives, or something that could help to bring the message to the community. Have you guys internally discussed how, other than the paper itself, how you're going to communicate with nephrologists and perhaps even patients about changing this reality?
Yeah, thank you very much, Roberto. This is a good question. And actually, I know there is here in France, with the French Society of Nephrology committee, they created a work group about this topic, about talking of conservative care, and more precisely about the terminology to use, because as you see, there is a lot of words that physicians use to their patient, and I think, and we think, that the patient not necessarily understand what does it mean, because there is a lot of terminology, there is a lot of terms, and that's very difficult for patients to do that. And in this work group, they have also included some patients to participate. So there is some stuff ongoing. That's a good point.
Thanks very much. Vivian, you didn't have time to mention, but I mean, did you look at the basic science literature for information that base the potential role of iron deficiency in the function of kidney cells that could be used as a background to your clinical study?
Yeah, hi. Thanks, Roberto, for your question. It's a great question, and we have discussed about that for a long time already. And actually, there are a lot of literature that shows the interference of even also iron content, in terms of higher iron content, leading to tubular lesion in the tubular cells, and as well as the possible role of mitochondrial in the kidneys, leading to this potential worsening kidney function through the iron depletion effect in these mitochondrial cells. So we still are looking for the best explanation to justify what we are looking for, but this is the main ways that we think it's associated.
Thanks so much. And Helmut, great to see you. And thanks for your presentation. One question from the audience here is about the introduction of the new non-steroidal MRAs in clinical practice. They're probably going to, come up very soon to clinical practice in Germany. Do you expect that there will be an increase in the use of the potassium binders with the introduction of finerenone, for instance?
Yeah, thank you, Roberto. Hard to tell at the current situation. I do not expect that the MRAs will take up pretty much in routine nephrologist practice. And as a matter of fact, I assume that this will not really influence very much potassium binding drug use.
Thanks so much. And I think we are coming to the, time to wrap up the session. I wanted to thank all three of you for the wonderful presentations and for this discussion. And I think with that, I wanted to, pass the mic to Bruce for his final remarks.
Great. Yeah, thank you. Thanks so much. Thank you, Roberto, and thank you to the speakers for this segment as well. Really just a ton going on. And once again, as we've noted throughout the program, please feel free to provide comments for us by the Q&A function, and we're certainly very, very happy to take questions after the session is over as well, just as noted here, but with Chris's response to the entire audience. So please feel free to reach out. Well, okay, and indeed, I think we will finish up on time. I just had a few closing remarks for today. I mean, hopefully what we have demonstrated is how rich the work is that we are able to do in this space. And once again, as noted, it takes just a body of investigators working with us and personnel. And actually for the team, do you have the slide with our collage to show? That would be great, demonstrating the worldwide reach and teamwork of the full group. Okay, there we go. Perfect. So once again, we are celebrating 25 years, reflecting such important input from so many people. It's really a team effort together with you. So just to very quickly review some, the 3 hours that passed from my point of view very, very quickly. We first of all had a session on HIF-PH inhibitors, and indeed 2 years after first approved in important Asian countries. And I really feel that we have not only with DOPPS, not only an incredible resource, but a real obligation to track and evaluate uptake and utilization of these therapies, because they really do have dramatic potential for quite dramatic input for dialysis patients, but of course in the non-dialysis CKD space, and for in-home dialysis, which of course we capture very, very well. So much more to come in this regard. With respect to the DOPPS sessions, I was really that were presented today, I was really struck again by the practice variation that we're seeing, which again, underpin the launch of our program 25 years ago. But we had a presentation showing quite dramatic variation in utilization of calcimimetics, for example, and separately in dosing of diuretics, which is good. That literally where European nephrologists are prescribing loop diuretics at doses probably 2 to 4 times higher than folks use elsewhere. So anyway, more to come in this regard as we identify and optimal practices moving forward. In the Pdops space, we launched our DOPPS Practice Monitor PD just a few months ago, and I think already are seeing some important findings, as for example, this clearly demonstrates disparities in utilization of home dialysis, particularly PD by race, just as one example. It was also striking in the PD section to realize how much there is still to learn. So for example, with respect to basic metabolic complications of advanced CKD, in the PD setting, there really is a need, opportunity, but need to understand better optimal practices. We then moved on to a really wonderful talk from Elliot Tanner describing our DOPPS ISN survey with a focus on lower middle income countries. And it would note once again that our data and others really indicate that mortality among dialysis patients with COVID is in the range of 10 to 30%. And this is really across countries, across regions. It's incredibly devastating diagnosis for dialysis patients. And of course, the trick here is avoidance of infection. And ultimately, as we're moving towards vaccination, we all need to do and advocate for reduction in inequalities and improvement in access to vaccines. And then lastly, the CKDOPS section I thought was also just fantastic, but I think pulled out a lot that we just don't know, right? Even though we might think we do, but we ultimately don't understand, as an example, optimal iron dosing in advanced CKD. Got great literature in the heart failure space, but not a lot in advanced CKD. So it's really useful to keep contributing to that work. The problem of hyperkalemia is still notable and substantial, very, very common. And as Elodie pointed out, conservative management, much more work to do in this regard. And more broadly, in the transition to end-stage kidney disease or to kidney failure space. All right, so all that said, once again, we really look forward to continuing to work with folks. Do ask people to reach out with research ideas and opportunities for collaboration. I would put in another plug for new countries, and we've just been so privileged to work with some middle-income countries around the globe. As examples, Thailand, Brazil, Gulf Cooperation Council countries, Saudi Arabia, UAE, and others. In all of those countries, we have just great examples of how we've demonstrated practice variation that probably could be improved upon, with quite rapid improvements in patient outcomes on that basis. Well, I would just to finish up, so we are now through 25 years, and indeed moving on to the next 25. So, all right, so another 25 to look forward to. In the hemodialysis space, we among many others, I think have done great work to help reduce mortality among in-center hemodialysis patients. That's the good news. The bad news is there's much more to do, as you all are well aware. And we're happy to be right in the thick of it and keep working with you on achieving better outcomes for patients, and not only survival, but better patient experience, quality of life for such patients. In the PD or home dialysis space, once again, perhaps one of the most important issues is assuring greater access and equitable access to PD, particularly with new payment models and new incentives that are coming to play in multiple countries, but in particular here in the US. This is going to be a very, very important time for home dialysis and watching closely what goes next. And then really, in the CKD space, two things. First of all, we're really focused on doing yet more to understand that transition to kidney failure. Much more for us to learn in this regard, and we've come up with some important ways that we think we can do yet better to focus on this space. And then secondly, prevention of kidney failure. And I think, as folks are well aware, with recent clinical trial data, there really is the possibility that we can make have an important impact with respect to truly reducing incidence of kidney failure. That's where so much of this the future lies. And actually, if you do some math fairly simply, you can imagine that we can make a real impact with some of these new therapies that have come to light. And as always, the issue is going to be application of those treatments, right? I mean, for the difference between great clinical trial data and great practice in the real world is often massive. And the trick is really to watch what occurs and ensure that patients are getting the best treatments that are available to them. With that said, I would like to wrap things up. Of course, we typically tie this meeting to the ASN, so I do encourage folks and to enjoy the ASN next week. The content, as always, looks tremendously exciting and informative. And then looking forward to next year, we will, of course, have our spring update meeting tagged to the ERA-EDTA and ASN, or autumn update tagged to the ASN. And our hope is that indeed we can have those in person so we can reconnect and strengthen relationships and make new ones as well. All right, well, this brings us to an end for the session. And once again, thank you for your participation, and look forward to much more to come. Thanks very much. Bye-bye.